Hello, good morning, everyone. It is my pleasure to start our first HUPO webinar and the Single Cell Initiative. Today we have an excellent speaker pool. We will have six people will talk to us in their advances in the single cell proteomics. We will have 30 minutes per speaker, leaving hopefully 5 minutes for the questions, but I apologize in advance that not many questions could be answered because we have a very tight schedule. I will try to get the few questions to be answered by each speaker, but since it's a webinar and we want to be on schedule, unfortunately, when the next speaker time will be going for the next time, I will try to go for the next speaker. Okay, our first speaker today, the Professor Matthias Mann. He got his PhD at the University of Yale in chemical engineering, and then after that, just he moved back to Europe, where he was a group leader at Amble University, and then later become a professor of bioinformatics in the South Denmark University. That's what I had my pleasure to meet him for the first time. After that, he moved to Germany at the Max Planck Institute, become a director of Max Planck Institute in Martinsville in 2005. And in 2007, Matthias became a director of Department of Proteomics, again back in Denmark, in the University of Copenhagen. Since then, I will not probably add too much time of looking at the many areas of the Matthias Mann group that been participating for the proteomics for the last 30 years, because otherwise Matthias will not have any time to speak. And that's why today we are going to hear what Matthias and his group are interested in single cell proteomics. And without further ado, please, Matthias, share with us your experience lately. Thank you.
Okay, and you need to participate to enable screen sharing. It's not enabled right now.
I'm not sure how to allow you to screen share.
Usually if you...
I think I allow you now.
Okay, now it works, yeah. Okay. Yes, so thank you very much for the invitation. I'm looking forward to share our work here with the HUPO audience. I'll tell you a little bit about our instrumental advances and then what we use them for in the direction of single cell proteomics as well as spatial proteomics. So, okay. So this is where we are. So my group is in two places, one in Munich at the Max Planck, where I'm right now, and then also in Copenhagen at the CPR, Center for Protein Research. So we are interested in the last years. Our interests have pivoted a lot, apart from the technology development, to actually applying it in clinical context, and that's also what gives direction to our technological development. So it's always with a view of how can we apply this to understand disease better or to actually diagnose disease. So we do a lot of body fluids, I won't talk about it, but also tissue. In a broad range of things like metabolic disease, cancer, and neurodegenerative diseases. So this is just overview, the usual things you have to do: sample preparation, mass spec acquisition, and bioinformatics. And as we go to ultra high sensitivity, all these three are, I would say, equally important. So that's become apparent through the work of many groups. And for our group, we've worked for many years and still work with a thermo platform depicted here, but also increasingly, and today's talk will focus on our work with EvoCep and the BUCA platform. And also for many years we did DDA, but we've now almost completely switched to DIA, as depicted here. So this is by way of starting with the with the bioinformatics. So I won't talk much about it, but we have a lot of activities, and some of them are relevant to today's talk. But this is also just a call out to if you're interested, you could look at some of our work, and particularly interesting perhaps the structural work and the deep learning algorithms here that we and others use to really get in many cases twice as much out of the data than we could before. But there's also a lot of caveats, as you know, and they need to be addressed, and that's by no means the case yet. So that's just the call out for that, and also another call out for review we wrote last year, where we give an overview of how deep learning in particular, or machine learning, can be used in proteomics, and we think we're actually only at the beginning of the journey, also to define biomarkers and such. So then, as far as the technology for us that makes ultra-high sensitivity possible, so that's been a fruitful collaboration with Bruker now for 8 years, I think. And there were several generations of students already that have moved on, but these are current students, and in particular Marvin and Patricia will talk a little bit about their work. So compared to the Orbitrap, we have this TIMS funnel here, where we stop the ions with a electric field that works the other way than the gas flow, and they're stacked up then here, and you lower this and you get the ions coming out in an orderly fashion. So this is shown here again. That's called the trapped ion mobility spectrometry principle, and so we store them here, push them over to the next part here, and let them out one after another. So then with this principle, when you follow that with a quadrupole to pick off the ions that are coming out at a given time, that's called passive principle. And this just illustrates in the ion mobility and M over Z plane how you can do DDA with that, like picking out different ions for no extra cost, so to say. So then you can do 10 or 15 instead of just one, or you can do PRM in a similar manner. But we do also in the single cells always DIA passive nowadays. So part of this alpha PEP universe that we're developing, they're developing tools for actually looking at the data, and I bring it up here because I think in the community we don't do this enough, that we look actually at the single cell data, because those spectra are more or less noisy, a lot of noise, and there's not a lot of signal, and we need to tease out the signal from this noise, but it's still good in our opinion to look at this, and for that you need tools. So we have this alpha viz that you can use, and we're also making it available for Thermo soon, where you can actually look at your data for a given peptide, and so this gives you summary statistics and look at critical proteins. And what's new, you can also now back project something you thought you should have seen in the data. So based on the deep learning, you can back project that into your raw data and see whether you can actually see it there, even though the software hasn't picked it up. So this is illustrated here. And we have cases in the paper, it's on bioRxiv, where we actually clearly can see it's there, but for some reason the software hasn't felt confident to call it out. So, and this is just the latest where we are with the scan molds here, and this is Patricia working with Bruker. So this is a particular way to do the scans, and I just want to emphasize here that you can actually get, together with the Evo set, we get 6,000 proteins even in 100 samples per day. So that's quite a revolution compared to a couple of years ago at least. So, and this works also for phosphor, and then we adopt this to phosphor cloud, and then we can get a high coverage, and it's also biologically important, and in a very short time we can get a snapshot of a whole signaling pathway, and we get phosphosites on proteins of interest like the EGFR receptor. So we can even get now 35 well-localized phosphorylation sites in only 21 minutes, and you could use this to see what the stimulus is doing in your cell, and you would have an overview of what generally EGF is doing on a cell from, in theory, only one 21-minute experiment, which would have taken I don't know, 15 years ago, it would have taken weeks and weeks. Okay, so now coming to, as you know, there's many different approaches to single cell, and we represent one here. So we've built on both the Bruker platform that we work with and also the EvoCell platform that we work with. So first the Bruker here. So to get really into the single cell where we only put one cell in, we reckoned we needed about 100 times more signal than we had. So, and then a factor of 5 came here from letting more ions in and some tweaks in the instrument from Bruker. That's now commercially available, that instrument. And then another factor 10 came from the EvoCell system, where I have a potential conflict of interest because I'm an investor. But this was developed for a completely different purpose, for clinical work. I mean, that it should run thousands and thousands of samples with one high pressure pump, but then we realized we can have this preformed gradient and we just push it out slowly. So for us, that's quite low flow, and you can do it even lower, as you hear later. But we push it out at 100 nanometers, and it's very, very robust. So, and then we get also for free, so to say, the very small reaction volume from the stage tips that are in this context called Evo tips, where we have the peptides here in a very small volume, like 20 nanometers, that we elute. And that works really well for us because you can just run one sample after another. So that's the concept altogether. So we put this into 384 place. For instance, if we have a single cell FACS sorted, because that's a small volume then, and we put it on, we digest it here, and then we put it on the, so there's no autosampler involved, that's very important. So we put it on the tips here, centrifuge, and then it's eluted here, and that would give us, as we publish, 40 single cell per day. And this is also what I said before, when we push it out slowly at 100 nanometers, we get this electrospray advantage of up to factor 10 here. And this is particularly a scan mode that we use here. So since the paper, we've made a couple of changes, but the major one would be that we now use the ion optics because it doesn't have dead volume on the spraying side. So it gives us also quite good peak width, even for the slow flow rates, as you can see here. And there's also mode, if you wanted to, you could have 80 single cells per day. So we have very low volumes here, and then this goes now into this commercial instrument. So that's what we do now, and yeah, it works quite robustly. So then when we did this for single cells, we can see that at least this rank order stays very constant, and even in the single cell it stays very constant. And this is for, we arrested in the cell cycle as shown here. So for interface cells, we got about 1,000 proteins here, identified and quantified in DRA, and but if they are arrested at the mitotic stage, for instance, they're a bit bigger, and then we got up to 2,000 proteins. It also made sense with the proteins that we identified. So if you do volcano plot, we find proteins that we know to be involved in the cell cycle already, or we can see by what's known about them that they probably are involved in the cell cycle. And this is something I think that's important for the or hopeful, I would say, for the proteomics community. Namely, if we put this together with similar data on single cells in this for these HeLa cells, we see that in the principal component analysis there's this funny shape, but what's important here is that the proteomics is quite separated from the RNA that you can also see here in the heat map and the cluster. But the cool thing is here that the CVs of the proteomics is much, much smaller than the CVs of the transcriptome. And that is because the transcriptome, most of the genes don't even have a single transcript because they don't need it. and that manifests in shot noise for the transcript, and we don't have that. So we have other problems, like we don't get deep enough yet, but biologically we are in good shape because we have this complete proteome. So that was for the single cell. Now moving on to the tissue, because we like to use this in the tissue context because of we think that's a very good place to look for good biology and good clinical use. So we have a long history in that with archived patient tissue and FFPE tissue, as you can see here. So this was a microdissection that we did before, and then we could find new biomarkers that, for instance, in this case, were in the stroma and not in the cancer cells themselves. But that's still thousands of cells. And what we, of course, like to do is we would like to pick out individual cell types in situ from these slices. So so that's usually also shown like this blender picture, that we don't want to have the smoothie, but we want to have the individual fruit. And I'll show you also in the end a cool application where that was quite revealing. So so yeah, can we do this then? And there's again many different ways to do this, as you'll also hear later. And you'd really want to do it in a pathology context. You really want to do this from FFPE tissue, because there's hundreds of millions of these samples, and they are quite well preserved even for decades. So that will involve laser microdissection, but also here, and this is a point maybe it's also not not universally appreciated in our community, is that if you're talking about single cells, you often have to say what single cells are you talking about. So usually in the proteomic community, we're thinking of HeLa cells, so it would be like this. But there's also the immune cells that we're quite interested in, but that's actually a factor 10 more difficult. Whereas, even we happen in our group have to worked with single cells even like 15 years ago, but these were eggs, right? So these are actually huge, and this could be done forever already. So you have to also think about the challenge scales with the inverse of the size of the cells. Okay, so this is now the deep visual proteomics approach. I've explained before, but I think I cannot explain enough because it's still not very clear. So we have the tissue here. So we have a slice. The slice is usually 2.5 micrometer thick, so you don't actually get a whole cell even. You get only part of a cell. And then you do one step of microscopy, and that's the usual pathology scanning microscopy. So that's very well established, but you still need the right markers to find the outlines of the cell, so there's a lot of work there. So it's not like cell culture at all. And then you use the AI twice. So once is to segment the cell, so find the outlines, and so we're going to use that for cutting out later. And secondly, to classify the cells into different classes. So that's illustrated here. So now it goes from one microscope to another. So this microscope can actually cut them out. And it's going to use the cell boundaries found here. And then we don't have technically single cell here, but we have the single cell type or single cell state. So these could be a couple hundred shapes that are equivalent to, like, let's say 100 cells. And then it goes into this workflow that I already explained. And our hope is that it will, for instance, for an oncologist, it will make a diagnosis easier, or see if the treatment actually helps. So that's the goal. And this was recently published in Nature Biotechnology. And these are some of the key people that push this forward. So here's an example. So we have the fallopian tube. It has two cell types, one to secrete the mucus, to keep the egg floating, so to say, and the other one to has cilia to actually move it down. They can be distinguished by whether they express this transcription factor. So Lisa here, she taught the AI in one afternoon what is one cell and what's the other cell, and then it cut out a number of shapes here. and you can see it cutting out there. It takes about a second or two to cut out one cell, and then when we do the proteome, it separates here like that. And this is the protein atlas from Stockholm, Matthias Ullén, and here we can see that indeed the FOXJ1 is only in our mass spec data when it's supposed to be there, and not in the other samples. And also these are other markers that we know from the protein atlas already, and it concurs with our proteome measurements. So this is to show we can also do this in cell culture, and that we can go not only to to single cell, but you can also go to just only the nucleus. Like here, we have defined different classes with different morphologies. You can see them when they're cut out, and then you can even learn something about what these different classes do. And the cool thing is that because we have this biological enrichment, the function of this class of nuclei in this case kind of jumps out at you. So for instance, this banana-shaped one, this banana-shaped one, it's actually involved in this EMT transition. And here's also a simple example from a rare cancer. So we want to just compare the cancer cells to the normal counterpart. We couldn't do this classically, even with microdissection, because here we have, here in the normal tissue, we have a rich morphology and different cell types, and here we have more or less just one cell type. So comparing the two in macro would be totally misleading, but we can now compare the two by DVP, and we get even suggestions for treatment here, like these highlighted proteins. And now, and this is melanoma, so it's more complex. So pathologist indicates different areas here, and we can follow, for instance, the melanocyte in these different regions. We sort of get a time course how the melanocytes evolved to the melanoma cells, even though it's a single patient. So we can do a whole experiment on one slide and on one patient, which also removes the genomic background differences between different people. So we think this is a very bright future, but it's also quite involved. So I'm joking that this one slide here, this unassuming slide, could be a PhD thesis. So this is shown in more detail. So we're moving in the direction of multiplexing here. These are just a few plex, but we have done now up to 21 plex on the imaging side. So you can define many different cell types. And that gives us a different, this gives us different classes, and we can back project, after the proteomic clustering, you can back project what these different classes actually do. So, and for instance, we can see the ones close to the surface, they do they're more immunologically hot, as it's called, and here they are involved more in degrading the matrix, and that prepares them for metastasis. So in closing, I promised you an actionable case, and this is spearheaded by Thierry Nordman from my lab. He's actually a dermatologist, and the case here is not cancer, but it's a drug rash. So people present, it's very rare, very rare, fortunately, but people do present even after aspirin with a drug rash, and then this can be benign or it can be very lethal. So not much is known about this. So when it's benign, and if you don't like these images, you should close your eyes for just a second. And so you would get these rashes, so they look terrible, but they're benign, and then there's the intermediate form, and then the more severe forms here. So for instance, there's this stress syndrome here, and then there's this TEN syndrome, which is actually very severe. So now you can open your eyes again. So what is it? It's rare and life-threatening, so it has a 33% mortality, but yeah, it could be even as simple as an aspirin that triggers it. And it's yeah, so the skin will actually peel in 48 hours, and you need to go to the ICR, and you have still 33% mortality. So, and there's no known treatment for it. So, and generally very little was known about it. So we have a small cohort here of the 3 different cases that I showed, or diseases that I showed you, or subforms of this acute drug reaction and the healthy. So we stain for cytokeratins, these are the normal epithelial skin cells, and we stain for CD45, that's the marker for immune cells. And this is just the normal DVP that we go through. So now this shows you, one of the samples. So we see here the keratinocytes here and the immune cells here. They're infiltrating. This is actually not known that they play such a prominent role here. And this is now cutting them out. So this is cutting out, this is the same picture or the same sample. So cutting out the skin cells and cutting out the immune cells. And these are the contours that we needed because these are tiny, these immune cells. And indeed, so these are 20 proteomes. So we always get two, right? The immune cells and the skin cells, as shown here, they separate as they should. And then this is very nice, I think. So if we do a principal component analysis, so this is in the skin cells where you might have expected the action happens, but actually in the immune cells themselves, the separation between healthy and drug syndrome is much larger than here, and also it's different. So this stress syndrome separates more from the other two, whereas this lethal separates far away here. So all this was not known because there was not much known about this. So then we do, and then it gets better because we can do, because it's proteomics, we can do functional clustering here. And you will notice here in this cluster, in this most severe syndrome, we see the interferon signaling. So it's here, it's the most prominent, but it's also here and in some other cases. So that's very prominent as a bioinformatic category. So knowing that interferon involves signaling, involves stats, JAK-STAT signaling. So we look in our data for the STAT signaling molecule, and it's indeed like tenfold more expressed than in the severe patients than it is in healthy. And now, and then we can go back and stain the samples for exactly STAT1, and then we see it in the immune cells, but also in the epithelial cells. And this is a simplified version of the signaling pathway, and the JAK-STAT pathway is known in dermatology. And what's even better, it's approved for another purpose, to use JAK inhibitors. And now that's what we can do, and we actually plan to do with the center in France. And the threshold to show clinical utility will be quite low because there is no treatment, right? So it just needs to be better than no treatment. So we are actually planning that. It may take a while, but we are doing that now. And this is just an outlook. So we're also trying to combine the two. So doing the single cell in tissue. So this is in the liver. Florian Schober was in Stockholm before. So this is 5 cells, but I can also just say that we've gotten it down to 1 cell now. And then, we have a review how we think this fits together with the spatial, single cell and spatial fits together. So you might want to read it. And this is a heads up for the last conference I'm going to organize in this series of proteomics conferences. Will be next year in Heidelberg in October, so you might want to make a note of it. So thanks for your attention, and I want to thank my group and a lot of industry collaborations such as Evosep and Bruker and Leica also. And these are some of the funding agencies and some of the collaborating groups. So thanks for your attention.
Okay, thank you very much, Matthias, for such a great talk covering many subjects. It's absolutely gorgeous to see where technology moved, and especially with the applications that you showed to us. With an interest of time, I have only one question here I would like to ask, because I think it's very much important question for the community. The question is, as you work with FFP tissues, how do you manage to extract proteins and get rid of the protein-protein crosslinks formed by FFP? Thank you.
Yeah, that's an important question. So when we started with that, and we're not the only ones, or even the first ones, we thought, definitely this could be very tricky, and also, like, even if we did this, then all these cysteine-containing peptides, they will be gone and so on. But that turned out not to be the case. So you do get a bit of a hit in peptide identification numbers, but no general damage. And we have actually published, for instance, in the Journal of Pathology, very streamlined protocol for how to exactly do this at a larger scale. So we and others have published protocols. So they're a bit harsh, but the proteins are the most stable biomolecules. So they actually survive quite well this treatment. But yeah, it's not trivial, but it can be done.
Okay, excellent, thank you. I suggest everyone just to read the article and see the already proposed protocol. And we're moving to the second talk today. The second talk will be from Erwin Schorf. He got his PhD in the Technical University of Denmark, and then after his PhD, he moved as a postdoc at the Copenhagen University for the Biotech Center. After postdoc, he returned back. Looks like he's back to his alma mater, and now he's associate professor on Technical University. He has a group of cell diversity lab. And please, Erwin, share with us what's your lab is doing currently now and how the single cell proteomics could help you to differentiate different type of cells. Please, Erwin, is yours.
Thank you, Bogdan, and thanks a lot for having me here today to share some of our recent work. I know my title was more technical, but I figured I would rather show today how we're applying single cell proteomics and actually also transcriptomics to gain a better understanding of human hematopoiesis. So basically today it'll mainly be two things: give you a brief background on the human blood system and single cell proteomics, and then spend most time on the actual case study of how we are currently applying it to human bone marrow. It's still work in progress, but we have some exciting results that I look forward to sharing with you. So for those of you less familiar with human hematopoiesis, basically we've studied this system for many decades, and there's a lot of great reviews if you want to learn more about it. But really, the early notion was that we have very distinct cell types, like the hematopoietic stem cells up here, and then the lymphoid and the myeloid populations here. And kind of the more we studied it, the more we learned about the heterogeneity that's inherent to this system. That indeed these hematopoietic stem cells actually consist of many different cell populations, and even further downstream, we have a lot of kind of confusing cell trajectories going on. And it wasn't until single cell transcriptomics that really the central dogma of hematopoiesis was changed to shift from having discrete cell states to actually being more of a developmental continuum down these different lineages, and that really only through single cell approaches can we tell what a cell is doing at any point in time in terms of what is it differentiating to, is it cycling, etc. And I mean, we're a proteomics community here, so of course we're also very interested in taking this further, but I would also like to use today to argue that we should learn from the transcriptomics community while we still can. And for example, this Azimuth reference map, I mean, Azimuth is a huge single cell RNA-seq resource. And in the case of human bone marrow, you can see here how 300,000 single cells isolated from human bone marrow look at the transcriptome level, from which we can learn a lot about what cell types are there and differentiation aspects. But of course, we then also want to investigate this further and really learn how do these findings from an mRNA space translate into a protein level aspect of these cells. I mean, we know that gene expression isn't directly correlated between mRNA and protein due to both translation and degradation, and especially these protein modifications cannot be captured with mRNA, but probably play a very important role in dissecting such complex cellular hierarchies. So to investigate this and to really be able to study the cell heterogeneity, we need global single cell proteomics approaches. And we advocate that, like everyone here today, that we need to use mass spectrometry to do this. And I will just put a shameless plug in for a recent review that was published last year, where we kind of try to summarize some recent advances in the field that's freely available online. So, I mean, I don't think I need to spend too much time here, but I think we all can agree that Bogdan Budnik and Nikolai Slavov really instilled a big momentum into the field with the original SCOPAMS approach, where we can use TMT labeling to basically multiplex these single cells first of all, for throughput. So now we can multiplex up to 16 single cells at a time, plus the addition of a carrier channel to facilitate peptide identification. And most people tend to use about 200 cell equivalent for this, but recent sample preparation approaches like the cell-on-one seem to be able to do with less. But of course, this does introduce certain challenges because this carrier signal in terms of reporter ion signal from TMT is much more intense than single cells. So we need long injection times to capture enough signal to get good enough signal to noise. But these MS2 spectra really are both for peptide identification and peptide quantification, and then each individual reporter ion would then correspond to a single cell. So we really try to adopt this technology early and make some improvements, some of which we published earlier this year in MCP, where we start kind of making more efficient use of our mass spec time through the use of real-time search. So we have the Eclipse in-house, which basically allows us on the fly to do an MS1. It sends the MS2 to a linear ion trap scan, which can take only 23 milliseconds to actually read out that MS2. And then it feeds every single MS2 into a real-time search engine. And then only those where it was able to identify a peptide, does it then select that same precursor for a very long and high resolution scan in the Orbitrap at either MS2 or MS3. So we're really using 750 milliseconds to collect enough ions. And just very briefly to show the impact of real-time search, is that now with using MS3 style, SPS MS3 style quantification, getting rid of most of the co-isolation effects, we're performing very similarly to the classical MS2 in terms of numbers, but now have much more accurate quant. But we were quite intrigued to see that our reticle approach, where we basically, again, do an MS2 scan, but now with much higher injection time and therefore higher accuracy and precision, we are able to outperform MS3 and classical MS2 in terms of numbers of proteins per cell. So with this workflow, we can now analyze about 1,000 proteins per cell at a rate of 160 cells per day with the latest 18-plex TMT Pro. We generate a lot of data. I think that's inherent to single cell. So Benjamin, a PhD student in the lab, spent a lot of time implementing a computational framework for this. So we called it Scepter, where we basically take both LCMS raw files, but also very importantly, the index sorted FACS files. So we use FACS sorting to deposit these cells, and we want to use all that data for later kind of cell sorting. And you'll see very clearly in the bone marrow data why this is important. So you can read everything about this in last year's paper, but basically the three main features are normalization. So when we go from each TMT injection clustering on its own, we actually get single cell resolution clustering out according to cell phenotype. We can do cell filtering based on two criteria, mainly number of proteins per cell, but also the sum signal to noise of each cell. So you can set cutoffs, and then to impute the data that's missing, which is also a big problem in single cell, we do k-nearest neighbor imputation. Why this is useful, I'll go very briefly through this. This is in the Nature Comms paper from last year. If we took this leukemia system here and we record all the FACS markers while we're depositing these cells, we ran about 2,000 cells here. You can see that we can start ranking these cells in terms of pseudotime, i.e. differentiation state, with the very primitive cells down here and the more differentiated cells down here. And in this system, the leukemia stem cells would define the most primitive population. And actually, we do indeed see when we overlay these FACS markers that that is exactly those cells that we're seeing down here, whereas the blasts, which are fully differentiated, are the more mature cell populations. So we can visualize the FACS data on all these embeddings. We can draw protein heat maps to really figure out along this differentiation trajectory of LSC to blast, which proteins are highly expressed or lowly expressed, and kind of how they're transitioning through these cell types. And we can really start teasing apart key differences between these cell types at single cell level. So we wanted to use this proof of concept work on a primary culture model on really primary samples donated by healthy volunteers. And this work is really the collective of Bo Porce's group, Fabian Theis's group, and a lot of PhD students across the labs to really try and maximize what we can get from this. So what we decided to focus on was the hematopoietic stem and progenitor cell compartment, i.e. kind of the upper part of the hierarchy. As indicated here on azimuth, we're not looking at the CD34 negative mature populations. And if you're interested in how to fact sort these, well, these are our gates and different markers that we use, but I'll get in a bit more detail in a second on how we use this information. But really just to stress that we're focusing on the primitive kind of stem cell and progenitor populations of this tissue. So Despina, she generated CiteSeq data on about 10,000 HSPCs from 4 different individuals using CiteSeq, and we found about 4,000 highly variable genes. Whereas Nil generated a large single cell proteomics data set on nearly 3,000 HSPCs from 6 individuals, and we recorded the FACS markers. We get about 3,000 proteins in total and around 950 proteins per cell. So an initial sanity check is always, well, let's plot this on a UMAP to see if we get cell clustering, and indeed both in the RNA-seq and the proteomics, do we get at least cell clustering, the cells of the same color clustered together, more or less, which tends to indicate that the protein level data we're measuring is telling us something about cell phenotype. But you might be wondering why some cells here are in gray. And that's because we annotate rather strictly as a first pass what cell is what cell type. And that's explained a bit more detailed here, where again, all gray cells were measured, but then based on their cell surface expression and the facts, we annotate cells differently. So we basically have this first CD34-38, where our stem cells are in the plus-minus compartment that we can then co-separate into 3 populations here, whereas this upper part can be segregated onto this plot here, and then we can further segregate on another level to really get all these cell populations. But we were setting very stringent gates. For example, up here, we're not calling these GMPs, but rather trying to get really that central part, or here with the MEPs, getting really a bottom part to be a bit more stringent up front, but of course with the overall goal of being able to identify cell types of all cells. And we'll get to how we do that in a few slides. So I mean, if another first sanity check would be, well, do our cells cluster according to what we know from facts? I mean, we know that a stem cell down here should be CD34 positive, but CD38 negative. And indeed, we can see that the CD38 expression of those stem cells is very low, whereas the CD34 expression is rather high. CD10, which is uniquely expressed on CLP cells, we can see that indeed that's only highly expressed on these CLP cells annotated in red. Again, seemingly suggesting that our proteomics level clustering is reflected back in the FACS data, which was very encouraging. So we can actually recapitulate this hierarchy. Then we figured, well, what about using this wide body of mRNA-based data to help us better annotate our populations? So for this, we used our CiteSeq data that we generated, but then annotated with the cell markers from azimuth based on 300,000 cells. And then first of all asked, well, of those markers from azimuth, which ones did we also detect at the protein level? And that led us to have this list of a few proteins that should, in principle, indicate whether cell is an HSC or a bi-ELMA or early GMP, etc. So then to first validate that these markers make sense, we first ported that back on the RNA level data. And then we saw indeed that, for example, an MPO, which we deemed to be a GMP marker, lights up very nicely in this purple cluster that we annotated through azimuth as GMP. For example, CLC, which is a bi-ELMA cluster, lights up here in this blue bi-ELMA cluster. So again, just confirming that the RNA markers that we are able to detect in protein level data do segregate RNA cells first of all. But how about protein markers? Because that's what we're doing this for, right? So our histone 1 marker was what we designated as an HSC marker, a stem cell marker. And indeed, we can see that in the protein level clustering, this is very high. The CLC was the bi-EOMA cluster I just described. And indeed, we see this cluster here. Based on the FACS data, we were calling these CMP, but probably there's a slight annotation problem. And this is a nice example of where we're learning from proteomics data how to better annotate our FACS data. And I'll get to that in a second. And maybe the last example here is MPO, which should be a nice GMP marker. And indeed, we can see that these pink cells here are nicely lighting up according to their MPO protein level expression. So the markers tend or seem to work out. So this means that we can use all azimuth markers that we detect in proteomics to actually start annotating all our cells. And we actually get much nicer resolution. So first of all, we are now able to annotate all cells, but we're even able to segregate certain cell populations, like, for example, these red CLPs actually seem to be 2 different cell types. So pro-B and pre-B, and then something that we call cluster 6, which is most likely still CLP, but actually it's already more heterogeneous than the FACS data would have led us to believe. Another very important finding here is that what we call CMP based on the FACS data, according to the azimuth markers, are actually bi-EOMA populations. And this is a hot debate in the hematopoietic field that do CMPs actually exist? Some people say it doesn't, and that they are already committed. And that's what our protein level data would actually also suggest, that they're not CMPs, but actually bi-EOMA. And the GMPs over here are actually also 2 subtypes. So we have GMP dendritic cells, and then the more classical GMP markers here. So we seem to be gaining more resolution from using these protein level markers based on azimuth readouts. So then, of course, you want to validate some of this, because maybe what you're predicting doesn't make any sense based on the FACS expression of the surface markers. And this is really what we're trying to do to convince the field of hematopoiesis, which has been around for many decades, that what we're measuring actually makes any sense according to what they've been measuring for all those years. So like I said, bi-EOMA is actually what we used to think were CMPs. Well, if you look at the CMP here, that were these cells here in pink. And we're seeing, well, actually, we just extended the gate a little bit down in that sense. So indeed, we're talking about the same cells in terms of FACS markers, but actually their identity is bi-EOMA rather than CMP. Our HSCs are following this same trend that they are in this population. And for example, the erythroids, which we used to call MEP, MEP is basically an upstream progenitor for erythroids. We can see that they fall exactly in the same set again as the MEPs do. So MEPs were down here and the erythroids are down here. So we're just allowing us or the data to tell us a bit more about what cells they are, but based on the FACS immunophenotype, these are indeed the right cells we're talking about. Like I said, GMP was divided into 2, and we can see that the main distinction is that GMPs are kind of down here, whereas these dendritic cells are more up here. And indeed, CD123 is a dendritic cell marker, whereas we actually isolated originally only this part, but you can see that indeed this whole part is more dendritic cells, and probably GMPs are better here. So we're really able to leverage this proteomics data to allow us to better annotate our FACS data. So the fact that we did both transcriptomics and proteomics, I mean, we now know that we can annotate our cells better in the protein space, but can we actually integrate these 2 data sets to create one bigger data set of basically 14,000 cells? And this is what Sabrina and Benjamin really worked hard on, to use a variational autoencoder in terms of deep learning to generate these RNA protein correspondences to help guide this integration, to then being able to do a joint embedding of protein cells and RNA cells. And lo and behold, after quite a lot of effort, this actually worked. And we can see that both RNA and protein level data, we see good clustering across this entire UMAP. So here we're just labeling the RNA cells with their corresponding color, and here we're just labeling the protein cells. So of course, there's a lot fewer cells here that we measured in proteomics, but we can see that, you know, what a GMP was deemed to be in protein space is the same as in an RNA space. So this is really something that we were very excited to see. And if we actually overlay them like this, we can see that we get full spread of all the cells across this UMAP in both protein and RNA space. And this allows us to start looking in much more depth what we can do with such integrated data. So we can really start doing this label transfer from RNA cells to protein cells and be much more confident about the cell type designation that we're interested in looking at and what proteins are actually driving these cell types. And that's something that we're evaluating now in a lot of depth. So I will just give you a few kind of highlights that we've managed to figure out so far. So again, we really like to use pseudotime to indicate a differentiation trajectory from primitive to much more differentiated cell states. So we have our HSCs here, which should be very low. And then we have our more differentiated populations kind of indicated in green and yellow. And then if we ask for specific populations, like for example, a GMP, how are cells differentiating from an HSC to a GMP? You can see a very clear trajectory here with the more or less fully committed GMPs up here. And this is exactly what we see in this map, that the GMP in purple are located right here. And now we can really start figuring out what proteins are changing across these differentiation trajectories. And same with bioma. I mean, there's hardly any trajectory here, but at least we can really do some differential expression analysis to figure out what made those cells different than the rest. So this was using CellRank, which was a transcriptomics specific tool published earlier this year, but we can actually use it on our integrated data to infer trajectories, and it seems to work actually rather well on this joint embedding and finding the correct end states. So the last thing that we're looking at a lot now that we have RNA and protein data is, of course, this never-ending question of correlation between these two. And this is what I just want to kind of show two clear examples, a protein called EF1A1 and MPO, which you might remember as the GMP specific protein. And then each column here represents the transition from an HSC to a bioma, or an HSC to a GMP, later erythroid, and so forth. So it's just following these cells along their trajectory. And we can see that, for example, MPO, which was a strong GMP marker, we can see both at RNA level and at protein level that they both seem to agree very much that MPO just needs to go up for a cell to turn into a GMP. But if we look at, for example, the bioma cells, and we have this EF1A1 cell, we can see that protein is very much suggesting that needs to go up, whereas the transcripts are falling along this differentiation axis. So how come that these cells or these processes are regulated so differently at RNA versus protein level? And that's something that we're trying to look into. Are there specific protein classes that are showing these trends, or what's actually really going on here? So I hope to be able to report on that in the not too distant future. So just to conclude, I mean, I hope I've managed to show that while it's still in its very early days, single cell proteomics is actually able to now start tackling biological questions, both of cell heterogeneity, but also to really monitor cell differentiation processes and cell decision making. Multimodal analysis, i.e. true multi-omics across the different modalities, is very beneficial. And here, as a proteomics community where we're still struggling to get a lot of cells through and very deep proteins, proteomes, and of course we just saw from Matthias beautiful work on how we're extending this, but we still have some way to go, I would argue. But that's where we should leverage the fact that we have very rich single-cell RNA-seq data to help us boost the data quality and interpretation of our single-cell proteomics data. So this integration is challenging, but I'm sure there's many labs working on this, and then early results seem to be very rewarding. So now we're really looking at differential expression analyses for the different cell clusters to figure out what protein candidates might pose as clear cell markers, and can we improve the FACS sorting strategies. And really with future aspects, I mean, we're of course wanting to expand this into the diseased state. So we have run a lot of AML samples, leukemia, or precursor states such as MDS, and we're trying to figure out, can we see how the cellular hierarchies are changing from healthy to disease? We're working on experimental approaches to be able to do RNA-seq and proteomics from the actual same single cells. We might hear more from Ying Zhu later on how he's actually tackled this challenge. And indeed, as we just saw beautiful examples of, I think the spatial single cell analysis holds immense potential clinically and biologically. So all efforts here are very welcome. So with that, I want to thank a lot of people. I mean, not just my group and Benjamin and Nil, who are joint PhD students with Bo Porza as well. Bo Porza was my postdoc mentor, as was John, where I started getting into hematopoiesis, but also Valdemaras and Susan and Pedro, who are the first group members of my group that I started earlier this year. We have some undergrad students that are really delivering great work. Fabian Theis, of course, for help with all the integration, and Sabrina, Thermo Fisher, EvoSep, Biognosis we're working a lot with now as well, the D2 Proteomics Core, and funding for keeping my lab going. With that, I'd be happy to take questions if there's time.
Okay, thank you very much, Arvin, for a great talk. I can see that you're already answering some biological questions, which is either wasn't clear before or new to the community. And I hope with time, you know, with all the technical improvements, we will build up our own azimuths and prove to the RNA-seq people where they've been wrong. And that's the target, to bring more proteomics knowledge toward the single cell type of biology. Okay, well, thank you for your talk. We do have two questions there, I think related to each other. That's why I'll read for you both. Could you please discuss your justification for any imputation? That's kind of one. And then the second question adds a little bit more specification to this. How was the missing values at the TMT labeled single cell? How did you adjust missing values of each channel for comparative quantitation? Yeah, please answer those.
Great questions. I mean, missing values are the killer, right? So if we measure one cell and we get 1,000 proteins, and we measure another cell, we get another 1,000 proteins, but the overlap is on a good day 650 proteins. So we need to somehow get better at measuring the same proteins and lots of same proteins across many cells. And I think the DIA field is going to be immensely important to solve this, at least from a data acquisition perspective. Because indeed, within a TMT batch, we're seeing very low numbers of missing values, but across batches, we are up at this 40 to even 45% missing values. So you then might ask, well, you have, let's say in this bone marrow data, 950 proteins per cell, but you're measuring 3,000 proteins across all those cells. So are you just imputing 2,000 proteins just to be able to generate these UMAPs on all of those 3,000? No, that would be highly biasing your biological interpretation. We set certain filtering cutoffs, and this is really project specific of, okay, how many cells do we need to have seen a protein in before we want to include it in our imputation? So for example, in our Nature Comms paper, we used a cutoff of 10 cells. So as soon as we've seen a certain protein in 10 cells, we start imputing it. But indeed, in the end, we probably have about 50-50 of real measured protein and imputed protein. But then for our differential expression and cell calling or cell ID determination, we don't use these imputed values. So it's purely for this visualization aspect that we use the imputation, not for any biological interpretation of differentially expressed proteins between cells.
Okay, great. Thank you, Erwin, for the answers. And we do have an extra minute. You have an extra question. Have you tried to separate the different hematogenies on the bias of transcriptomics and proteomics data? Thank you.
I'm not sure I understand what hematogenies are.
Then you're the next level from me. I definitely don't.
No, I mean, yeah, I can answer similar. I mean, we're seeing similar separation between the two. We had hoped that we could find more cell populations in the proteomics data because we expect more resolution. But I think here it's this fact that we're only quote unquote measuring 1,000 proteins, that we're not getting to the key cell, differentiation proteins like transcription factors, epigenetic regulators. And I think that's where we can really gain a lot of strength once we start picking up those types of proteins to really show where RNA-seq is getting it wrong, and where protein level data will really help better resolution.
Okay, well, thank you very much, Erwin. We are up with time. We're going to the next speaker.
Thank you.
Our next speaker is Roman Zubarev. Roman being my lifetime mentor, and he got his PhD in Uppsala University in Sweden, moved to the postdoc at Cornell University, and came back to Europe, become a junior professor in Odense University. That's where he brought me as a young student to the field of the proteomics, which I didn't know anything about. After that, he moved back to Sweden and became professor at Uppsala University. Currently, Roman is the head of the department at the Karolinska and the molecular biometry group. Please, Roman, share with us your experience with the single cell proteomics at your group.
Thank you very much, Bogdan. Actually, my talk today would be more about chemical proteomics, but also the lessons that single cell proteomics can learn from chemical proteomics. So what is chemical proteomics? It's a proteomics-based methods of identifying drug targets and mechanisms of drug action. There's a lot of need, there's a great need to develop new drugs because the efficacy of many drugs is low. And so most drugs are small molecules and they bind to protein targets. And when they bind, they are pretty promiscuous, and more often than not, they bind to several targets at different concentrations. So there's a need to identify the true targets and the off targets. And that's what we're doing with chemical proteomics. People bring us drugs or molecules, prospective drugs, and drug candidates, and we don't do any chemical modification to these drugs, and that's very important. So we treat the cells, and then we try to identify the target, the binding site, the mechanism of action, the mechanism of cell death, changes in the redox state, and since recently we started to identify also or determine the time, the residence time. And so why does chemical proteomics need a single cell proteomics? Because the cellular response to drugs is heterogeneous. Of course, there is single cell transcriptomics, is very well developed, has an advantage of PCR. I wish we had PCR for proteins. We don't have it. And so we cannot go that deep, but with the process, biological process dominated by the protein production, then there is a good correlation between mRNA and protein levels. And I would say you don't need single cell proteomics in these cases. But in a steady state, this correlation breaks down. And in these processes dominated by protein degradation, such as cell death, there is no correlation at all, nothing to be expected between mRNA and protein levels. So if you try to merge the proteomics data with transcriptomics data, it's going to be very confusing. And this is what is happening in the chemical proteomics, where most drugs just aim to kill the cells, like anti-cancer drugs. And we're using 3 tools, basically. One is based on protein abundances, another is based on protein solubility, and the third one on the redox state. The protein abundance tools is the oldest, and it's based on the empirical observation that in late apoptosis, the proteins that change their abundance most are the proteins that are related to the drug mechanism or drug targets. And so to add the specificity, we use a panel of drugs and sometimes a panel of cells. And OPLSDA is a great method, bioinformatic method, to purify your data when you contrast the proteomics change in one drug against the proteomics changes imposed by all other drugs, and the proteins which are upregulated or downregulated. These are the ones that are usually containing the targets. And so we designed a tool which is called Protein Target Miner, and designed a library of 57 drugs that represent all known mechanisms of action. There are about 13 different mechanisms of cell death. And so in PCA of the protein changes after 48 hours, we got 13 dimensions. It fits very nicely. So here you have examples of the proteins that are definitely targets. For instance, methotrexate. When you look at the methotrexate, you see the D-HPP is highly upregulated. And so you can bet your farm and your house that this is a target, and it is indeed. Paclitaxel is tubulin depolymerization inhibitor, and the proteins which are specifically upregulated are tubulins, and in vincristine is tubulin polymerization inhibitor. So the proteins that are downregulated specifically are beta tubulins. So they are the targets of the drug, and so on and so forth. So we put a tool on the web. Anyone can download the data, their own data, or merge without data. There's a data set there and process the data. PLSDA analysis is done automatically online. So we wanted to know whether the same approach can be translated down to single cell proteomics. There's a paper published this summer in Analytical Chemistry. We started with methotrexate because we know D-HPP is behaving very well. We looked at how the cells behave upon treatment with methotrexate, 3 hours, 6 hours, 12 hours, 24 and 48 hours post treatment, and we could clearly see the cell population separating. We wanted to know which cells decide to die after 48 hours, because the dose was such that it was LC50. Half of the cells going to die after 48 hours, but half of them are going to survive. Which cells will decide to live, which cells will decide to die? And so, and whether we could determine the D-HPP to be a highly upregulated protein, one of the most upregulated proteins after 48 hours. And you see, indeed, even at the single cell level. So the quantitative aspect of single cell proteomics wasn't clear to us. Was it quantitative enough to apply the methods of chemical proteomics? It turned out that yes, they are. So we can differentiate the cells that are deciding to die and the cells that decided to survive early on, about up to 12 hours, when you couldn't differentiate it by FACS. And I would subscribe to the idea that if you can differentiate the cells by FACS and do it, and then do bulk proteomics, you will have much more accurate and deep analysis. It's only in the case when the FACS methods do not separate cells, then single cell proteomics makes sense. So we tested the conclusion that single cell proteomics is quantitative enough to determine the drug target at single cell level with two other drugs, camptothecin, that downregulates its target topoisomerase 1. You see, after 48 hours is one of the most significant proteins, and tomudex with targets thymidylate synthase, and again, it's there. And so what if we apply our OPLSDA analysis for these three drug data set, single cell level, and you see methotrexate shows that D-HPP is the most upregulated, one of the most upregulated proteins when you contrast methotrexate with two other drugs. And here you have camptothecin, top one, is one of the most downregulated, and tomudex, thymidylate synthase. So it all works and nice and dandy, and that's all very well. However, the protein abundance is just one dimension in proteomics. And what we learned from chemical proteomics, that it is insufficient. More dimensions are needed. What kind of dimensions? The one that provided by so-called thermal proteome profiling is very tempting. In thermal proteome profiling, here you see Misha Savitsky. He was also a PhD student in my group once upon a time, and in a group of people who came up with an idea to treat the proteins at different temperatures, and then look at the soluble fraction, and then find the melting curve and how this melting curve shifts upon drug addition. The idea is that the protein that is interacting with the drug will shift its melting curve the most, and it works very nicely. And actually, this method now used very widely, and we apply this method to determine the enzyme substrates, but also very easily you can determine protein-protein interactions. You just take a cell lysate and add a protein of interest, recombinant one, in excess, at least tenfold compared to the endogenous level of that protein, and look at how the solubility of other proteins changed, either left or right, up and down. And most often than not, the protein that change significantly their solubility will be the ones that are interacting with the added protein. And we show that. And we could also determine the enzyme substrates in the method we call SIESTA, system-wide identification of enzyme substrate by thermal analysis. And there you add to cell lysate either vehicle or cofactor or enzyme or both. And the idea is that since the enzyme needs a cofactor, it is when both are present, the thermal shifts should be the highest in true substrates. So you need to do some kind of filtering, but when everything's said and done, you will get pretty good results. That's all very nice, but in order to apply this method to single cell proteomics, we need to increase the sensitivity at least tenfold, because you need to analyze 10 different fractions treated at 10 different temperatures. And then you have to face another issue, which is surface binding, and also low concentration. At low concentration, the proteins don't want to aggregate. And therefore, something needs to be done to bring, to bridge this gap and bring TPP closer to single cell proteomics level. And we were thinking that integration of the samples into one could partially solve the problem. So instead of taking 10 different samples and analyzing them separately, we would just merge them in one sample and analyze it at once in the method called proteome integral solubility alteration, or PISA assay. Works like a charm. And you can merge it with the previous method based on the expression analysis, T-text, and you can do both the solubility and expression analysis at once. And we applied this method to stem cell differentiation when we took fibroblasts and converted them to iPSCs and then differentiating them to embryoid bodies. And as a control for stem cells, we use embryonic stem cell line. And as a control for differentiated cells, we use RKO cells. And then we did both expression proteomics analysis, all these states, as well as solubility analysis with PISA. And what we found is that when we do PCA, both these dimensions give almost equal separation. So you can merge these data. They are complementary to each other, and you can create a pseudo timescale. And with that pseudo timescale, we determine the most important proteins that seem to be contributing to this differentiating state. So how to translate PISA to single cell proteomics? Well, it works better. PISA is much more sensitive than TPP, but the surface binding issue remains. And especially as we found, the concentration issue. At low concentration, the proteins don't want to aggregate. They need other proteins to form clusters. So the sensitivity of PISA is not as high as required for do single cell level. We'll try to find other ways to aggregate proteins, and one of the ways is to add some salts from what is called Hofmeister series. That's a paper published in Analytical Chemistry this summer. So what we found that certain salts, such as zinc chloride, they actually allow proteins to aggregate at room temperature. You just increase the concentration of this salt. And but in order to, you cannot put this in samples together, because then the concentration of the salt will average out. And so what do you do? So we found out that you can add another salt, and they make zinc salt insoluble, and then that doesn't affect the proteins, and they stay soluble, and that allows to use methods similar to PISA to integrate the samples into one. We call it iPISA for ion PISA. And we show that this allows one to reduce the amount of sample use, because one can use an analysis method, sample preparation methods that are borrowed from single cell proteomics, that is preparing samples on glass slides. And we could do analysis using less than 1 microgram of protein. It works very well. But the drug binding is not the only parameter of interest in chemical proteomics. It turns out that it is the residence time of drug on target, which is much more significant because it has better correlation with in vivo efficacy of the drug than just simple binding. We decided to try to see whether we could use PISA method to determine the drug residence time. This is a paper which has been accepted to analytical chemistry yesterday, so it should be online pretty soon. And what we're doing, we can work with either cell lysate, or we can work with intact cells. So we treat the samples with a drug, and then after some time of incubation, we remove the drug, and then we wait and wait and wait. And as time goes, we take aliquots and do PISA. And then we see how the protein solubility, the target solubility changes. It goes to background level, to the level of untreated sample. And what's the time scale? We started with simple molecule uracetamide. We all use uracetamide, or most of us use uracetamide to alkylate cysteines covalently. And so we thought that all proteins will show curves which don't decay, but it turns out that while many proteins show really these curves that don't decay, but many proteins also show curves that decay pretty fast. So uracetamide makes not only covalent binding, but also non-covalent, and then we clearly see this difference. Then we go to ponatinib, which is a kinase inhibitor, and try to see what it does. It also supposed to be a covalent inhibitor, and we found that it stays for a very long time with the target lean, but for other kinases, it may not stay very long. You know, it may stay just several minutes, half an hour, and so on and so forth. So that allows us to actually rank our targets. First of all, the good target will bind the drug very well and changes solubility quite significantly. That's one parameter. The second, it should bind it at low concentration. So we can use concentration dependent PISA as a second dimension. And as a third dimension, we use the time of binding. And when you rank your targets by these 3 parameters, you see that some of them are really good, such as lean or Mark 14, and some known targets are not so good. And that's very important to know for drug development. So the conclusion is that chemical proteomics reveals drug target without chemical modification of drugs. And because of cell response heterogeneity, emergent chemical proteomics with single cell proteomics is beneficial. The method we call FITEXP or pro-target miner use expression level changes for drug target deconvolution, and it's already translated to the single cell proteomics level. PISA performs target deconvolution and ranking based on the solubility changes. It is not yet translated to the single cell proteomics level due to the sensitivity issues and others, and we're working to resolve these issues, but we're still not there. The ion-based PISA reaches sub-microgram level of sensitivity, and it's our hope that this method will be translated finally to single cell proteomics. But of course, as always, more work needs to be done. I would like to thank my group members, former to the left, and Bogdan is one of the first, if not the first, PhD student, as well as current group members, funding agencies, collaborators, and thank you very much for your attention.
Okay, thank you very much, Roman. Great talk. I do have comment that it was very interesting to see that some of the cells make a decisions to die upon the time very early. And I think it's very much important to highlight that fact that we do have a situation where we can actually define the situation where we can treat with known drug and see those cells that actually become very quickly drug resistant and try to fight with that, which is something that we would never see on the bulk proteomics. That's why I see with the power of the chemical proteomics going towards the single cell. This is just my comment, and we do have a first question. What are the limitations to study PTMs with chemical proteomics? Is the dynamic more important?
The PTMs.
Yes.
Yeah, actually, yeah, I think so. I think we need to measure the occupancy. For occupancy, we need to know the abundances of both modified and unmodified peptide, and that's a challenge, because if you use the enrichment strategies, then you lose the unmodified part. And if you don't use enrichment, then you may not see the modified part. So that's a challenge. But I would like also to say that we found that most modifications change the protein solubility. That's what makes PISA so attractive. Whatever happens to a protein can be translocation or attachment of a chemical group or a tertiary structure change. It most often, not always, but in many cases, it changes its solubility, while the abundance may stay the same. So indirectly through PISA, we measure the integral PTM state of the protein.
Yeah, that's actually was my probably comment on that. When you do the integral, you kind of already assume that those PTMs that would influence the abundance of unmodified protein would actually be in that integral already kind of calculated. But you never try to do the PTMs separately and see how the say only those PTMs that you see in the normal analysis without any enrichment would differ picture upon the drug treatments versus the just normal proteome.
No, no, we don't. We didn't. But I'm sure that maybe Matthias, somebody else may from the panelists can answer this question better.
We do have actually a couple of minutes left before our time for the next talk, and I would like to have a live conversation if anyone would would like to participate in answer to that. What's the general view of the PTM analysis versus general proteome? Would, in your opinion, change our knowledge for the proteomics, or you think the analysis of the normal PTM, normal proteome without PTM would already be sufficient to kind of answer questions in the drug discovery field?
Well, the PTMs are great, but there are 300, 400 different types of PTMs, and therefore the answer should be specific to PTM. We've been interested in such modification as deamidation that leads to isoaspartate formation, and developed methods for analysis, mass spec-based methods, as well as immunology-based methods, and link that the modification to Alzheimer's disease, and try to see whether we could add something to the mechanism of Alzheimer's disease, and turns out to be quite possible. So yeah, so PTMs are great and very important. In terms of drug development, I don't really know. I mean, again, the answer is should be specific to PTM. Of course, if you use a kinase inhibitor as a drug, then what you're doing is inhibiting phosphorylation, and sure, so PTMs that's what you're doing. You kill the cells through PTMs.
Okay, Matthias, do you want to add anything?
Well, I cannot really add much on the side of the drug development, but one common question is, when can we do PTMs in single cells in general? I think it would actually be a very nice feature, and it would be something that certainly single cell transcriptomics cannot do. So but we think it's maybe more in, for instance, in the DVP context, that you could also in the future study PTMs. If you really study only a single cell, it's gonna be a while until we have that sensitivity, right? So it would be maybe more in the spatial proteomics context. It could actually add a lot, and that's something certainly that we are trying. But for true single cells, it may be a while. But I've said these things before, so maybe it's sooner than I think.
I would like also to add that what was great with single cell proteomics with Advent, thanks to you and Nikolai, is that it's not only single cells. It just highly sensitive, ultimate sensitivity of proteomics got a boost, and we're applying single cell proteomics methods to the case when we do have many cells, but not millions, right? So small number of cells, same methods, and it's really great.
Yeah, I actually would like to share to the larger community your definition of such proteomics, especially I think it very much applied to what Matthias shows later in his talk, that we are rising onto the few cell proteomics. And I think the few cell proteomics given quite a fruitful answers. And those few cells could be achieved by different methods, by fact sorting, by other type of a priori knowledge about the cells kind of specificity, and get a couple of those cells together and do the kind of classical proteomics, so to speak, but with much more sensitivity, gives us much better result. And I can see this across other groups is growing. That's why few cell proteomics is on the rise. Okay, I think we are very close to the time. Unless any other questions, I don't think any other questions shows in the chat. Thank you very much, Roman, for your talk. And we're moving to the next speaker. Our next speaker would be Claudia Crotecta, and she is getting. She got her PhD in Vienna in a group of. There and in molecular pathology in Vienna, and then recently this year she moved here to Boston to the Broad Institute as a postdoc, and she continued her study of the single cell and now across the ocean. And I would like to hear what's kind of new and advanced techniques you apply, especially in the area of the single cell preparation. And please share us news from Broad. Thank you, Claudia, to be here.
Thank you. I'm gonna try to share. Can you see my screen? Perfect. Okay, thank you very much for having me today. I think I'm gonna switch a bit the focus of the talks that we have heard so far, because as usual, I'm gonna do it a bit more technical and a bit more not so much on the definitive application side, but also something that we find very interesting in our samples, and how we actually try to do the entire workflow and really focus on very detailed analysis, and also to really report whatever we see and whatever we find most challenging, also in the data processing and data interpretation aspect. So let's get right into it, if that works. Yes, so today I'm going to be talking about multiple different aspects of the ProteoChip workflow that we have implemented already back when I was still in Vienna with Carl, and how we have been developing it so far, and why we're actually spending so much time on our single cell sample preparation workflow is that obviously we are dealing with a lot smaller specimens as in standard workflows, and we really want to have the most streamlined workflow to process our specimens as reproducible as possible, and then introduce it all the way to the mass spectrometer. And in the last couple of years, a lot of different groups have started to work on improving sensitivity of current workflows. And they find a lot of different smart ways how to overcome peptide losses and different limitations that we were facing with standard sample preparation and acquisition processes. And we have therefore currently a variety of options that we can choose from, including the obvious multiplexing, where we have started with Nikolai and Bogdan, or the more label-free area that Matthias also, for example, now is focusing on. And all of these different combinations have their individual advantages and also disadvantages, on which we and also a lot of others have done a lot of benchmarkings and comparisons and really detailed analysis on what is essential to really take care when doing these analyses on a single cell level. But anyway, today I will talk mostly about our take on label-free single cell proteomics and how to most efficiently deliver our peptides into the mass spec. And for this, as most of you know, we have teamed up with Selenium to optimize the entire workflow, and how they're doing, how we're doing this is we're using their piezoacoustic dispensing platform, the Selenium that you can see on the left-hand side. And what is really the most interesting aspect for me is that this instrument actually allows us to do not only piezoacoustic droplet generation, which allows us to minimize our sample preparation volumes to picoliters, nanoliters, whatever you desire, but it also combines image-based cell isolation within the instrument. So based on shape, granularity, and size of your cells or cell types of interest, you can select your cells within the Selenium and basically dispense it into any kind of array that you're analyzing. So a lot of different groups have developed different systems how to process the samples. So we have, for example, the nanopods that Ying and Ryan are probably going to talk about, also now implemented with the Selenium. Our proteochips, and also Nikolai is his own workflow also implemented within the Selenium. And after we do isolate our cells of interest, we do perform temperature and humidity controlled incubation within the instrument. And that allows us to really eliminate all of the sample transfer processes and sample manipulation processes to really focus on reproducibility. And what we wanted to do with our proteochip to really focus on is also the direct combination to the HPLC that we're using. So we really aim with all of our designs to be able to interface with the HPLCs. For the label-free chip right now, we use a sort of 384 well format, which is very easily compatible with all kinds of different standard autosamplers. And our workflow really aims at focusing on the three main aspects that I find most important for single cell sample preparation. And that is starting with miniaturization, that we have reduced our sample volume to around 40 nanoliters, which is quite challenging for a lot of HPLCs, but when we perform the injection, more and more, it's getting more and more easy to really inject those very low volumes and keep peptide concentrations as high as possible to minimize losses. Then we have really focused also on the automation. As I was saying, we perform everything on deck on the Selenium. And I'm very happy actually that Bogdan keeps the webinar very short, so I can run straight away back to the lab where my single cells are currently incubating. And then I will just insert my chip into the HPLC, and off we go for a couple of more injections. And now for our current projects, we have decided to really completely focus away from the multiplex sample preparation to our label-free samples, which also means that I'm currently mostly working on the timsTOF SCP, which overcomes a couple of problems that we were initially facing with standard isobaric labels. But obviously that massively inflates the numbers of analytical runs as we have to look at the cell one by one. And to still conserve the reproducibility, but conserve the heterogeneity that we're actually dealing with and that we want to analyze in single cells, we want to analyze the samples as reproducible as possible and minimize missing data within those extremely large cohorts. And how we do this is DIA. And after Matthias's talk, I don't think I really need to go into too much detail on how DIA works or how timsTOFs are improving all of this. But very briefly, DIA obviously reduces missing data by overcoming precursor stochasticity. So we aim at very regularly isolating all precursors individually, all precursors equally across the entire sample cohort. And in order to be sufficiently fast, we need to widen our isolation windows when performing DIA. This obviously in turn also results in a lot of co-isolation of multiple precursors, which makes these spectra inherently difficult in comparison to DDA. But this is where the amazing timsTOFs comes into place, where we add the additional layer of ion mobility separation, where we continuously concentrate and pre-separate the ion packages within D2 with an analyzer number 1, and while we're continuously eluting ion packages from the analyzer number 2, we can already accumulate the upcoming ions in order to not have, in order to be as reproducible and as sensitive as possible. And also this allows us to not only separate our analytes based on their precursor mass and their retention time, but we actually additionally acquire a very unique parameter, the ion mobility. And then we have not only separation of otherwise co-eluting precursors, which we can see here. These 3 precursors would have otherwise been co-isolated. Ion mobility separation allows us to really distinguish those 3 precursors, but that does not only allow us to quantify actually all peptides that we're subsequently including into our analysis, but we have 3 parameters of these peptide moieties that allows us to improve identification characteristics and also really be sure whatever we're analyzing is true. And with this in mind, we really wanted to optimize the methods that we're using within these new instrumentations. So we noticed that not only we have to tweak our DIA passive methods to the individual PTMs that we're analyzing, but we also need to really focus on the most productive areas that we want to cover within this MRZ and ion mobility range. And this really changes when we analyze very low input or single cell samples. So we here see, let me see if I can put the on. So we really wanted to focus on the most productive area here indicated in gray, and we tailored all of our DIA passive windows to really cover that area most efficiently. And together with Sebastian, who currently works at Bruker, good for them, we really optimized a standardized way how to place these windows to cover as many precursors as possible. So in our current method, we we use 8 cycles to really be as fast as possible to still cover over 95% of the precursors that we would be interested in. We perform these cycles as short as possible with around 100 milliseconds ramp time, because we use a very short, efficient gradient, effective gradient to be increasing our throughput while we're analyzing these cells one by one. And to really allow for proper quantification and a minimum of 5 points across the peak in our very short gradients, we do perform, as I said, 100 milliseconds or lower of ramp times with 35 Thompson isolation windows. And in contrast to many of the methods that have been postulated so far, we actually do split our ion mobility range in order to separate these precursors out even more. Now, let's go into the data and to really answer or try to answer the first question that I usually get from my colleagues. How will you analyze all of these files, and how will that actually impact my processing of my searches and the computational resources that we have in our group? And our initial options for all of our timsTOF DIA data was Spectronaut, which we still currently use, mainly because the interface is very practical and the data can be easily explored. So in a bit more detail, Spectronaut integrates Pulsar for DDA searches and also to generate experimentally derived libraries. They also have more recently implemented a library-free approach, which they call direct DIA, where you overcome basically the very lengthy need to generate libraries that are experimental specific and may also not really represent the heterogeneity of those single cells that we are dealing with. As an alternative, we have the famous DIAN, which is now also integrated into FragPipe to really seamlessly combine MS-Fragger for DDA searches and also library generation and extremely high throughput DIA searches. And even though Spectronaut and DIAN allow you to basically interface with pretty much any experimentally derived spectral library, the most convenient options are, as I said, Pulsar for Spectronaut and MS-Fragger for DIAN. And most importantly, FragPipe is proposed to run on a desktop computer, and the timsTOF raw files are getting smaller and smaller. We know that. But especially when we're dealing with these very sparse single cell data, it really still takes ages to analyze all of these files. They create huge intermediate files, and my laptop is usually not very happy if I urgently need to compute something to give a talk on. So what I was really happy to see is that in our group, there was continuous developments on how to streamline that and really outsource basically the computational leads. So Mani and Koi from our group started to implement both DIAN and FragPipe into Terra, which is a Google Cloud-based platform that is very easily accessible via your browser, and you therefore actually really don't have any local hardware requirements, and you can still massively parallelize your analysis. And what I really personally like about this is that you can share your data, your pipelines, and also your intermediately processed files with any of the other users within the Terra network. And what you do is you compute online, and then basically you just download the files that you're interested in. This obviously doesn't overcome the problem. It mainly relocates the difficulties, but is better suitable for very impatient people like me. And I would like to usually look at my data in real time. And this brings me to my last option on how we do analyze our TIMS DIA data in real time. That is the Pacer box, which allows you to process not only DDA data using their integrated search algorithm on their Pacer box, but it also now integrates a TIMS DIAN option that basically processes your DIA files on the fly and retrospectively can completely be integrated. And matched between runs. So basically all of the options that are available in DIAN are also possible to perform on the Pacer box, except the library-free searches that are not currently completely integrated. And since the PACER runs on an external computer, it doesn't actually occupy any of the computational resources that we have in the lab. It basically just requires the addition of a computer that sits right next to the timsTOF table and can be very easily accessed from pretty much anywhere. So these are the three options that I would like to introduce you today, and also how we apply those to our single cell data. So here I'm going to show you proteins and precursors identifications from single cell injections that we have searched against a single shot DDA library. So what that means is we have basically prepared all of our samples in exactly the same way for the DDA injections and the DIA injections. We have prepared the DDA library and then use this exact same library just for comparison. For a matter of comparison, we have used the exact same library for all of these four search algorithms. And we have sort of scrambled them around as well, and we see similar trends. So what we can see here is with the search using Spectronaut, we identify Spectronaut and DIAN, the most recent version of DIAN. We identify around 1,000 proteins per single cell injection, and that is without match between runs. Now, when we look at an earlier version of DIAN 1.7, which is the basis for timsDIAN, we see that we see a slight drop in identifications both on the precursor and the protein level. And then when we perform analysis using timsDIAN, we see an additional drop. We do not know yet what are the real identifications based on these samples, but I really want to highlight that here we're dealing with the exact same raw files, just analyzed with a different software. And you really have to be extremely careful what kind of numbers or what kind of peptides you report when you're dealing with these extremely sparse data sets. Now, we really wanted to find out what is the main advantage or what is the main difference between those search softwares. And we started by looking into their activity in actually preserving precursor profiles across a large cohort. So here I'm showing you 50 analytical runs on the x-axis, and on the y-axis we see the percentage of missing peptides. And what I hope you can see, even though the blue and the green is not very easily to recognize, I see now, is that DIAN and timsDIAN actually do make a better job in reporting complete precursor profiles. So we do have very similar numbers of missing data when we look at all three of them. However, Spectronaut has less of lower complete profiles as DIAN or timsDIAN have. And just to make this completely clear, this is the exact same data that I was showing previously. It is just displayed in a slightly different way, still reporting the same exact conclusions that I've been observing within these data sets. And what I really want to make clear also is that even though we only see small differences between the three, we see that in timsDIAN, we actually recover almost 10% more peptides with a complete profile as compared to DIAN. Now, we also wanted to see how accurate these identifications are across the platforms, and not just by identification on an identification manner, but also on a quantification manner. So here I'm showing you the quantified profiles of TIMS DIAN versus DIAN. We see a relatively acceptable correlation between the two of them, which is not utterly surprising since TIMS DIAN is based on DIAN. However, when we look at the lower so in the lower abundant area, we can actually see that these profiles do diverge more. So we do see something that we expect: the more abundant the more intense our profiles are, the better they do get extracted and quantified across these platforms. That becomes even more apparent when we look at the comparison between Spectronaut and the DIAN versions. So we see Spectronaut here on the y-axis, and here we see DIAN and TIMS DIAN. The correlation does actually drop. It also goes in line with Q values. So the lower the correlation between the search algorithms per se, also the lower confident those identifications are. So we really need to be very careful in what to report, and also how we quantify or we extract those quantification profiles. And to look into a bit more detail, and after a great discussion that I had on my last big talk here in Boston, we wanted to see how accurate those identifications are. And for this, we did not only generate experimentally derived libraries, but also predicted ones to actually really calculate or estimate the actual FDR when we try to trick the software is actually in making mistakes. And to look at the different aspects of library searching and the confidence of identifications that we're generating, we generated three different libraries. One with the standard human FASTA input, as you would expect to perform your analysis. Then we artificially inflated our database by just basically doubling the FASTA file and adding additional decoys to it, where we end up with only a fourth of true positives within our FASTA file. And then the third one, where we really wanted to see the combination of humans versus non-human, a non-human organism, to basically supplement a decoy organism that is approximately the same size of the human proteome, and to really look at the true FDR. And I'm only going to show you today results from Spectronaut and DIAN, where I was mentioning that TIMS DIAN is not fully implemented for its predicted libraries. So we start to compare basically what we did as a sanity check. We just analyzed against a non-human FASTA file, and we were happy to see that there were almost no IDs. So that's reassuring somewhat. And then we searched against a human, the human FASTA file, and we recover very similar numbers of proteins and precursors compared to the experimentally derived library, even though we see in DIAN, we actually see an increase of almost 20% of protein identifications when we use the predicted library versus the experimentally derived library. Now, when we look at the artificially inflated library, we basically actually identify the same numbers of proteins and peptide precursors. Even though we have extremely inflated the library size, we see only a drop of 7 or 8% respectively for Spectronaut and DIAN. However, when we then combine our human and non-human library and look at the actual FDR, I was quite puzzled to see that with Spectronaut, we report a FDR of 0.1%, which is great. However, with DIAN, we're more in the range of like 3%. And I think this really indicates that we really have to be careful what we report in these very sparse data sets. And also what I've been showing last time, and just want to make sure that the overlap between these softwares is not great. And even though I'm always reporting 45% of strict peptide sequence overlap between two search algorithms, this holds true for pretty much any search that we do. This we don't know yet where those reduced overlaps are coming from, but I think this is this really opens up the questions and opens up a lot of different areas that one can still explore. I nevertheless want to show that both of these search algorithms do actually span 5 orders of magnitude within our single cell data. So the overlap is not great, but we do get very comparable results from both of them. And just the last teaser basically is that we not only want to combine our proteochips with standard HPLCs, but we really also wanted to push on throughput again. And for this, we have teamed up again with Selenium and with Avosep to push the limits on throughput and chromatographic separation time. And we have come up with a new proteochip design that directly interfaces with the Avotips. And to demonstrate really briefly why this is extremely important and why I'm always an advocate of no manual sample handling, because humans are usually the problem. When we see the transfer, the manual transfer, we identify around 500 proteins. However, when we do the exact same prep and do the direct transfer, we see an increase of more than 50% in protein identifications. And also we are looking at the CVs where we can see massively improved reproducibility whenever we're not pipetting at all. And what I'm very glad to show you already that we have also teamed up with Ion Optics to really reduce the measurement time by more than 50% again, and perform the 80 samples per day method. And I'm very glad to tell you that we actually do recover similar dynamic range, even though we're analyzing them at extremely high speed. And I hope to show you more on that in the future. And I'm very briefly going to wrap up with what I started. So I really do think that there's a lot of options out there, and a lot of them have their individual advantages and disadvantages. But we really are very happy with our current labor-free proteochip sample preparation in combination with DIA. And we currently focus a lot on chromatographic separation. And I think this is really also an area where there's still a lot to do, which basically brings me to my last point, where I think the most work is still to be done, which is data analysis, data interpretation. And really, I don't think that there is a right way to answer all of the questions out there, but we're getting there, and I think it's a very interesting field to be in, and to yeah, I think every project has its own advantages, disadvantages, and also really thoughtful selection of whatever you want to go for. And with this, I'm at my huge acknowledgement slide, where obviously I'm very happy to be now at the Broad, where Steve and Namrata are giving me my new home with all of my team staffs, and I'm very grateful to all of the great collaborations that I'm happy to have with these amazing people. And if there's time, I'm happy to take questions.
Hey, thank you very much, Claudia. Really great talk, interesting coverage for the sample preparation. I think it's very important message that you just sending right now to the community, that you already put together something which is available already on the market as the solutions. And those solutions can be put together from different vendors to go basically human-free workflow. I do not too much believe in the human free workflow, but when you put things together as a humans, then they usually work better further down. We don't have much time. I would like to basically quickly go through the questions that we have from the audience. The first one for the isobaric labeling with proteochips and cell and one, can you use organic solvents like acetylnitrile but as a droplet generator at a picoliter level? If so, where you can find a good example that are reproducible and accurate outcome?
Yes, so the current workflow that is provided by Selenium actually does allow you to perform organic solvents dispensing. This is actually not a problem at all. We have come up with a very fancy solution on how to really make this work very stable. They also have now in the newer version a automated workflow that allows you to basically really be quite fast in labeling 16 and 18 plex, all diluted in organics or even DMSO if you feel more confident in DMSO versus acetonitrile.
Okay. Unfortunately, the last question due to the time limits. Did you compare the proportion of proteins identified based on one peptide versus those identified with more than 2 peptides using the different softwares? Probably related to the ratios between, Spectronaut and Diane, those that are outliers. Are those with 1 peptide or not necessarily?
Yeah, so we in general eliminate one-hit wonders because we really notice that additionally inflates the variability between the softwares. So we are very stringent in whatever we're reporting, and I do see actually a lot of differences in the scoring, and we're still trying to really make sense out of what we see or where these differences may arise. If you have any suggestions or any ideas, what you would be interested in looking at, you can contact me anytime. You will find my email online. So I'm happy to discuss offline.
Okay, there is one more about LFQ single cell, but I think, please contact directly to Claudia with the questions. Due to the time limitation, we would go for our next presenter. Thank you, Claudia, for your excellent presentation. Our next presenter, Yin Zhu. He got his PhD in Zeeland, if I'm pronouncing correctly, sorry, University, and then moved here to the US to the PNNL, Pacific National Northwest Lab. As a postdoc, he was there in the different positions, ending up with the senior research scientist. Recently, Yin moved to the Genentech as a senior principal scientist to proceed with his ability to prepare single cell proteomics. And I'm happy to have him here today to represent his advanced preparation technologies for the single cell proteomics that they developed at the Pacific Northwest. Please, Yin, stage is yours.
Thank you, Bogdan, for your introduction. And it's great to be here. And so I recently moved to Genentech. But that was all my talk actually was done in PNL and Amazon in the lab. So today I will talk about how we can improve the single cell proteomics and the tissue protein mapping based on the ion mobility filtering, and you see the FAMs provided by Thermo Fisher. Okay, later. Okay. So as we know, single cell proteomics is a quite new research area, but really on this direction, the whole proteomic field actually have done a lot in the past 10 or 20 years. So we can clearly see at the beginning, we use a very big column, right, around at the mil per minute. So we slowly reduce the flow rate to microliter per minute. We call it macro APLC, and nano APLC is commonly used for all the proteomic assay mostly. And because they reduce the flow rate, actually we also develop the nano spray, and we also show by reduce all the elect spray flow rate, we can get a very high ion transmission from liquid phase to gas phase. So also you can't go the matching of the flow rate or nanoliter scale. But if we look at sample prep, we do have some improvement, but it's not too much. So we most time now we are doing microliter sample prep. So usually use the tubing or use microwave plate. So also it's also matched to use the microliter sample prep and the nano RC to do analysis. But we really see, usually we prepare microgram of sample, but really each nano RC will only run 100 or 200 nanogram of sample with most of the sample. So that's often what we see in the past 5 to 6 years, the big boost in sample prep by reduce the sample prep volume from the microliter to pico to nanoliter scale. And we use the example of nanopod, but really not only the approach available now, it also have many different technology available, and we see today. So the nanopod we working together with Rankeli in PNL is the when we reduce the sample volume to a small droplet. So we actually just fabricate the chip and to generate hydrophilic spot on hydrophobic background. Then we do everything actually in this small droplet here. So because the volume is so small, we have to build a robot, as you can see here, to do reagent dispensing and to enable us to do nanoliter sample prep. So what do we call it nanopod? Because we also do everything in one droplet. So we do not transfer them between different reactor, or we do not do like precipitation or purification. So in the video show you to not only, because we built a robot, we not only can do the sample prep in the small droplet, also we can withdraw back to inject into LC-MS. So we do a lot of the evaluation, and we really we learned in the past several years is for the low sample input of single cell proteomics, the miniature reaction is really critical. So not only we see at the beginning is use a small volume, we can boost the protein ID by reduce the surface loss and get high sample recovery. And also it can, the small volume actually enhance the protein and enzyme concentration. You can get much higher digestion efficiency. In terms of the labeling, you get a similar boost because you also increase the reaction kinetics of the chemical labeling. So another two benefit is when you do the very small volume reaction, you don't have to do sample purification because the volume is so small, the contaminant from the reagent or from some actually is very, very low because you reduce the total injection volume by 1,000 times. So it's actually very important for single cell study because if you want to run 100 samples, you want to get high robustness or LC-MS. So the reduce the total volume also help that. And use the microfluidics, you can do photolithography. So you can get a very good design, like the next nanopod we developed last year. So we can get a parallel sample preparation, get a high throughput sample prep and analysis. So today I will focus on the mass spec side is the introduce a new approach we developed for use ion current for single cell proteomics, MS1 ion current for single cell proteomics. We like it because the MS1 spectral, you can get very high resolution if you use OB-trap or FDRCR. So at such high resolution, you get pretty low residual compression. You can get a very good quantification. And because because there's MS1 is a lab-free approach, so you have minimal batch effect if you run all the samples together, all easy to correct. And also for multi-omics, you also want to get the absolute quantification. So Usually we do this before it use RBAC. That's also based on level 3 MS1 approach. Finally, because we do not rely on MS2 for quantification, so we can use the algorithm like AMT, accurate mass tag, or magic run. This is based on retention time and M2Z to do the identification. So it can get less miss value because you don't have to rely on all the MS spectrum. So the major question is how we can use the new approach to increase the protein coverage based on MS1 our current protein analysis. So there's actually there's a lot of study have focused on this, how to improve the protein coverage based on MS1. So this first study is done by Pasatolik and Dick Smith in PNL. So they developed this dynamic range enhancement applied to mass spectrometry. The major idea is called the DREAMS, is to address the challenge of dynamic range. So dynamic range is because all the mass spectrometry have the limitation of the charge capacity. So if you in one scan you have very high abundant ions, you cannot see the low abundant one because you just quickly fill the trap during accumulation. So idea of DREAMS is to dynamically include the high abundant peptide in the next scan. So in the next scan, you only focus on this low abundant species, so you can get a very high sensitivity detection because it only accumulates this low abundant species. So another approach by participant group is the boxcar, is also try to address the dynamic range. So the idea of boxcar is to fragment the ion into many different populations during MS acquisition. So if you have many fractions, you likely separate all the high abundant and low abundant in different scans. So you can get a very good detection sensitivity for low abundant ones. So we are wondering, can we use FAMS? As we know, FAMS is also fragmentation technology. So it's a filter each time you use one compensation body, you filter on one ion populations. So either we do a test. This is typically single cell signal. If you look at if you do any single cell, so commonly if you do not use any ion filtering or ion mobility, the majority of the ion you see actually is charged one actually from the air, as you will see 445, or from the contamination from solvent. Because it's high abundance of this charged one, we call the contamination ions. So we can quickly fill the trap. We only get a very low ionization time. However, if we use the FAMS, by remove all these charged one species from air, from solvent, we get a very long injection time. Also, because we only focus on this multiple charge peptide ions, we can get a very good signal-to-noise ratio for all these peptide ions. So this actually shows what's the difference if we use FAMS or non-FAMS. So standard, as you can see, when we run single cell, most ion injection time for MS1 is only 30 milliseconds. But we use FAMS, you can get 250 milliseconds for each scan. So you can accumulate a very long time also for peptide detection. So that's why we developed this, we call it TIFF, is transferring identification based on FAMS filtering to boost the sensitivity of the mass spec detection. So how we do that? So we first sort the cell during the cell isolation use FAX urosaline 1. So we sort 50 cells or 100 cells in each sample. So we run these samples through individual survey of FAMS. So we can get, we call it spectral library. And for each identified peptide, we associate with not a 2 tag, there is 3 tags, is CV and the retention time and M2Z. And for single cell, we run the single cell actually use multiple CV. So we get all this, a lot of scans. And then we use mass quant or use mass Fragger. So we can do the 3-dimensional matching by matching all CV and M2Z retention time, then get the peptide identification. So that's the two major advantage of 3-dimensional matching is because we can get a very high injection time for MS1, we can get a high sensitivity. Also, compare with AMT or match between one, the three-dimensional M-1 feature matching also reduces the false matching. So here is the typical, we see, sorry, it's very small, it's very hard to tell. But what I want to say here is a typical FEM scan. Usually we can use too many CVs if we use short gradient. Usually use 2 or 3 CVs. So we have one MS1, many MS2. And about for TIFF scan, it's very different because we allocate most of the time on MS1. We have very long MS1 scan and 1 or 2 MS2. It's very few MS2 scan in each cycle. So as you can see, it's the typical FEM scan majority of time spent on MS2 to get a high coverage. So the TIFF scan, the majority of time actually spent on MS scan to get high sensitivity for single cell. So we try to compare what's the difference of the fault discovery rate based on the 2-dimensional or 3-dimensional matching. So we just generate a spectral library contain both human and bacterial peptide. Then we try to look at if we only run human, no any bacterial peptide in the sample, how much bacterial we can matching to is a kind of false matching. So here data show the peptide level. We have 4% of the missing matching. Is actually the Shrinala is the bacterial peptide. We should not see them in the human sample, right? But if we use a 3-dimensional matching, we can reduce only 2% of the false matching of the data. So at the protein level, we can reduce from 10% to 5%. So also we want to look at the quantification at the single cell level. So we do something very similar. We have both mouse and bacteria of the for spectral library. And for sample, we have generated 2 different samples. One is the mouse peptide with very low amount, 10 picogram of bacterial peptide. Another sample is the same amount of the mouse, but that's 3 times more of bacteria. We want to see, like for mouse protein, we should not see any difference because there's the same amount of sample. It's clear also that for the chain is exact as 0. And for the bacterial peptide, we know it should have a 3-fold difference. It's also quite exciting. We see even we do not have a MS2, but only better MS1 3-dimensional matching, we still can get a very good quantification. Most of the protein identified as a 3-fold chain. So here is the data we compare our previous, and here is the user TIFF. We can get an average about 1,200 protein, use the label-free approach, and compare with the first single cell study we do in 2018. So we can get more than 1,000 protein from the data set. That's very encouraging of the improvement of sensitivity. So because there's a lot of push in single cell field to do multi-omics, most of the data, most of the projects are using the sequence approach, right? They use the sequence to look at the epigenome and genome, or epigenome and transcriptome. So because we can do single cell proteomics, we want to know if can we do single cell multi-omics by combining sequencing and mass spectrometry. That's what we focus on in recent years and try to develop a new technology for both parallel transcriptome and protein analysis for single cells. So how we do that? So we call this new technology is nanosplit, the nanodroplet splitting for linked multi-omics investigational transamples. It's a kind of variation of nanopod. We still use the same nanopod chip. So what we do is we first sort the cell on the single droplet and then put the surfactant to release the protein and RNA. So next, what we do is we align this sample droplet chip with another chip. We call it a septal chip. It only have buffer, no any sample inside. And then after align them to mix the 2 droplet array and then split them. So we can, in theory, get the 50% of the both RNA and protein each droplet. And then we have one chip for RNA sequence, another chip for proteomics. Here is the video. Hope I can play on it. Okay, so here as you, so we have one chip is already sorted single cell array. Here is we have 48 of droplet and 48 single cell in the chip. So we put a spacer between them. So they're easy to control the distance between the two chips. And then we put another chip, it only have buffer. So here we just use 5 millimole trace. So you know any samples in this chip. So what we do is manually align them together and then push the spacer to allow these two droplets to merge together. And then after 10 seconds, we just release. Because the droplet is so small, it's very easy to get a very good mixing between them. So here is the data we get. So we use SmartSeq tool to do RNA sequencing because it's quite easy to do and also is compatible with our low throughput workflow. As you can see, even after remove the gene and below 5 rate, we still can get like over 5,500 of gene detected with RNA sequencing. And so also we can see the CV is lower at the 11 cells and become higher at the single cell because of high variation of the single cells. For proteomics, we use our mobility-based chip approach. So as you can see, for single cell proteomics, we can get 800 protein from the same single cells, and we'll get higher if we use 10 cells, 3 cells. Also, we see the similar CV as shown by Matisse talk is we get much lower CV for proteomics because protein is more stable in cells. So here is just a quick demonstration. We can see the different two small cell line. One is C10 is epithelial and civic and endothelial cell is kind of model cells in our study. Here is the clustering of two modality, two cell type. Here the red one is proteomic, and this blue one is here is the RNA sequencing. As you can see, the cluster based on the modality first, and then each cell type also cluster together. It's quite exciting. And we first compare the correlation between the same modality. As you can see, we can get higher and lower sequencing is as expected. The most exciting thing we see is because we do the same RNA from same cell, we can do inter-cell correlation between the two modality and also intra-cell. In theory, we can should get a lower, we should get higher correlation between the same cell, right? But really, we do not see the big difference in inter-cell and intra-cell. It also tells us it is really hard to interpret the RNA sequence data and protein data because even in the single cell, same cell, they do not have very good correlation. So also, I think there's still a lot to study here. We still need to spend time to look at what's going on with this data, but really something you surprised to see. So another we want to move is the field also have bigger interest now is moving from the single cell biology to spatial biology, because if you really go to clinical, all the clinical people actually every data look at is a tissue sample from a patient. For single cell proteomics, there's also other limitation. Its practical view is pretty challenging to get a good single cell suspension if you want to do patient sample. And also when you do the dissociation to get a single cell, you can lose a very important information of the cell reorganization and also where the cell come from, where is neighborhood of these cells. And because you also do digestion, you lose all the important information of extracellular matrix protein and also secreted protein from the cell, how it affects the tissue microenvironment. So that's why there's a bigger interest in spatial omics now. So Ran Kelly and I start to coupling the spatial technology with nanopod. So when we choose is ZEISS LCM system, it can do pretty good, very precise dissection and collection. So the major, I like it because it allows you put the chip very close to the tissue to collect the tissue. So if we do very small dissection here, as you can see here, we do 20, actually here is 50 by 50, only have 5 to 10 cells. So after dissection, we direct collect into a small, into each droplet to do some prep. So it's a very slow process, but it's quite compatible with LC-MS because LC-MS actually is slower. So we can do all the collection and we can get a near single cell resolution, 20 micron of the tissue dissection collection. So recently we are focused on is how we can get a high coverage at the high resolution. So that's what we are doing, is coupling the TIFO approach, because TIFO we can get very good coverage and with the LCM. Here, what we do is we, as an example, because we all know the islet have very important function in the human tissue. So what we do is just look at what the different islet, single islet here is the blue color, and then look at what's outside of the islet. It's kind of demonstration of the technology to see if we can see the difference between the two major function unit. So use the TIFO approach, we can get very good coverage at it's not single cell, it's about 5 to 10 cells. We can get 23,000 of peptide from each pixels. And it's about 3,500 protein. And in total for this, the total 48 sample, we can get 4,000 proteins. Because we can get so deep coverage, so we can use a lot of tools developed for spectral transcriptomics to do spectral network. Spectral network try to look at the relationship, the gene co-expression between the different locations. So it can clustering all the distinct spectral map together. So as you can see here, we use the Ghetto provided by Guo Chenyuan in the Sinai, Mount Sinai, and to do the spectral network analysis. As you can see, we can get the 3 very different of the, even we know that should have only 2 different function unit in the single islet and adjacent samples. So but we see 3. So one is islet high, is the protein enriched in islet, is the endocrine of the cells. Also, we see antenna high is antenna cells, and as you can see at the 3 here, we also see this gradient across the from top to bottom area. It's kind of interesting why this happened. So after we do the reactor analysis of this spectral variable gene, because we get a set of gene for each, so we can clearly see the islet high. We can see also insulin processing signaling by insulin receptor, and also you can see here is 3. The antenna high is more like a digestion function. So for this top-down gradient is also interesting because we most of involve the mRNA splicing, and our cell cycle, it kind of tells us more oxygen enriched or nutrition enriched in the top area. So yeah, as a summary, in this study we show the user mobility of FAMs. We can improve the MS1 based the protein quantification. Also get very high coverage, get good accuracy. And we also show clearly we can identify now in model study we see today is we can reliable quantify 1,000 protein in single cells and single pixels of the tissue sections. And also present our preliminary data how we can extend single cell proteomics to single cell multiomics by the split the cell lysis into 2 different reactions, so we can do different assay or metabolomics, transcriptomics, use the nanospeed technology. And also we are very excited to further improve the spectral proteomics of protein mapping for pathology to get more insight of the disease. So finally, I want to thank model work done in PNL. So Ran Kelly and Dick Smith, we are working together to build the nanopod platform. And I collaborate with Liliana Patatolik after became individual PI. So all the work are done by the postdoc here, and Juming with about single cell, James is about multiomics, and Yumi focus on spatial, and also Sarah and Andrea support the whole group. And also we have also great collaboration with all the teamwork in PNL and collaborators. And okay, so I'm close my talk. I'm looking for your questions.
Yeah, thank you very much, Ian. Great talk. Thank you for representing Nanopods workflow for our audience. And hearing your latest move towards the multiomics is something that I think the community should pick up more and just go towards. We are ourselves interested in this approach. You do have a question from the audience. For the spatial analysis, do you randomize the sample analysis in order to identify run order effects?
Yeah, that's a great question. We do have to randomize. And so after we do the dissection, when we run the LCM method, we just randomize all the sequence, because for the tissue, actually, you clearly see a lot of the gradient, because some location you only have one cell type, some location have another cell type. So you have to do the randomize. You do not want to see the batch effect between the two different populations.
Okay. Due to the time limits, we have to move to another speaker. Thank you very much, Ian, for your talk. And I'm glad to introduce our next speaker, which will be the last speaker for our webinar today, Ryan Kelly. Ryan Kelly, he got his PhD in Brigham Young University, and then after that moved to the PNNL as a postdoc and then research scientist, senior research scientist. Currently, Ryan moved back as associate professor to Brigham Young University, and he still has an appointment in environmental science division of the PNNL. I do believe Ryan will present to us today like a more democratic version of the nanopods that a larger community could use for the introduction to the single cell proteomics. Ryan, please, it's yours.
Thank you, Bogdan, for organizing this. I've been really excited about the other talks that we've been able to listen to. I love just that the frontiers continue to expand. There have been a couple of times in the last 6 or 7 years that I've been working on this where I'm like, well, are we about done? But it's clear that there's just so much more to do. Things like the developing towards these drug development, drug discovery pipelines. And I also really appreciated the highlighting of the need for improved sensitivity, because that's mostly, we'll be talking about kind of the analytical workflow and data acquisition workflow that we've been working on recently. So as Ying mentioned, we were working together and early on developed this nanopods workflow. And I think sample preparation was the right place to start, because just the fact that we could miniaturize the prep and achieve improved coverage while reducing the sample input by a factor of 500 really showed that the sample preparation was the bottleneck. And of course, as Ying mentioned, we extended this by coupling with FACS, and more recently with the Cellin one, we extended the nanopods workflow to the single cell level, and couple with laser microdissection for spatial proteomics. And I think this has all been well covered. And what we have generally seen, I think as a community, there have been efforts to further miniaturize sample preparation down from a couple hundred nanoliters to 10 nanoliters, and that has benefits, especially for pooling TMT based samples. You don't want to inject such a large volume, but in general, we're not seeing much additional gain from further miniaturizing down to, say, picoliter. It looks like we're doing fairly well, and in some cases, we can actually relax the volume requirements without taking too much of a hit in our overall sensitivity. So, given that foundation, we're thinking, how can we further improve sensitivity, measurement, throughput, quantitative accuracy, and accessibility? And so won't get too much more into the sample preparation, but we'll talk a little bit about separations, where we're working there, and MS acquisition and data analysis. So we're really obsessed with reducing the flow rates of our analyses. And I think this, the results of this direct infusion experiment, where we're just looking at our sample, our mass spectrometer signal as a function of how many analyte molecules we're introducing into the mass spectrometer per unit time. And you can see here at 300 nanoliters per minute, which is where a lot of proteomic experiments are operated, setting the LC flow rate to 300 nanoliters per minute. If you reduce your flow rate down to around 20 nanoliters per minute, you get 10 times more signal per analyte molecule, at least with our analytical platform that we're looking at. And so even going down to 100 nanoliters per minute doesn't give you a huge benefit in terms of sensitivity. And we really see this exponential climb as we go to lower and lower flow rates. So that's some of our motivation for going super low. And the way we achieve these low flow rates with LC-MS is by simply reducing the column bore of the LC column. So T. Trong in my group is really great at packing these narrow bore columns. And so by reducing from a typical 75 micron ID column down to 20 micron ID column, we can maintain the same separation characteristics. And the main thing that changes is the flow rate that we're using for this. So typical column these days in our group is 30 centimeter long, 20 micron ID, packed with Dr. Maish 1.9 micron particles. Although this is still a challenge for our group. So a lot of our daily work is still done with 30 micron ID columns that operate more like at 50 nanoliters per minute. So when we're really trying to push the limits, we go to these 20 micron columns. You can see with a 20-minute elution window, we about this is our MS2 spectra, our PSMs, our unique peptides, and our protein groups identified without any match between runs. This is just 1% FDR and MS2 based identification. And we'll be talking about how we can increase this. And the separation's pretty good, not quite as narrow a peaks as what Matthias was reporting. And I think that's because we do not have an integrated emitter yet for these home packed columns. We're just using a zero dead volume connection between the column and the emitter. And so we're probably giving up some of our peak capacity in there. And that's an area that we're working on. So those work well. We get great sensitivity, but there are challenges associated with these low flow separations. And I'll talk about how Xiaofeng is working to address these. One is that with these high pressure, low flow connections, if you have a leak, it's really difficult to find where that leak is. So you're messing up your analysis, but your ability to remedy that is difficult. So we found ourselves just saying, I don't know where it is. Let's just rebuild the entire plumbing of our system. And then that in general, active peptide elution is only one portion of the overall duty cycle of an LC-MS analysis, right? We need to regenerate the column. We have this gradient delay. Peptides then elute, and then the column needs to be washed to get rid of, high high hydrophobicity species. Sample loading is in there as well. And so as you go to really, really low flow rates, your active portion can become a smaller and smaller portion of the overall duty cycle. And now we're letting this $1 million mass spectrometer sit idle for a larger and larger portion of the time. And we don't like that. So kind of inspired by the Jorgensen's group with and developments with offline gradient generation, followed by isocratic elution, and of course the EvoSep approach that uses this offline gradient generation as well. We're looking at that and looking at how we can use this. And one benefit of that is that we reduce the high pressure connections, right? There's a lot more low pressure portion of the plumbing network than just a few high pressure connections. So Xiaofeng is working to kind of take this and multiplex this develop multi-column LC systems that allow us to be, you can see over here, once we get the first sample loaded and analyze, eluting, we can be eluting one sample here. So we've loaded a sample into this low pressure gradient with the gradient behind the sample. All right, so we have just a tube with our sample up front and the gradient behind it. And then while that can be eluting, the isocratic pump can be regenerating another column. And at the same time that's going on, we can be eluting at low pressure and a higher flow rate with a binary pump from a trap column into this storage loop. And then while that's happening, we can be loading sample number 3 into the storage loop. So it's just kind of trying to find out what things we can do at the same time. And there's always some concern when you're working with multiple LC columns that the data you get from each column is going to look a little bit different. And we're concerned about that as well. But this is, working at 30 nanoliters per minute, continuous peptide elution from one column to the next, and they look pretty good. And in terms of proteome coverage that we achieve, here we're working on a 30-minute cycle. So this would be a 48 sample per day method. And you can see this is just plain old data dependent acquisition with an Orbi trap. But you can see we're getting about 1,500 proteins per say 2 cell sized aliquot, and then with match between runs that goes way up. And you do see one column performs a little bit better than the other, and so we have a couple percent difference, kind of goes up and down. And so if we're really worried about that much additional technical variation, we can always segregate our studies and be sampling from one chip with one column and another study corresponding to a different well plate or chip for the other column. So we can run 2 studies overlapping, but keep them analyzed on separate columns. And so that's some of what we're working on the LC space. We would love to go to even lower flow rates, but there are additional technical challenges that we face in further miniaturizing our columns until we can go to some sort of a open tubular approach. And then the final area that I'll be discussing in terms of the workflow is the mass spectrometry acquisition and data analysis, right? So when we're analyzing really low input samples, including single cells, and this is sort of specific to Orbitrap instrumentation, but it takes longer injection times to have an adequate ion population to have a productive MS2 spectrum, right? So if we try to go too fast, we don't have enough ions. The MS2 spectrum is quite low quality, and we may not get a match. So that leads to fewer overall MS2 spectra that we collect. But but if we're waiting to collect these long to collect enough ions, we might as well run the Orbitrap for MS2 in a higher resolution setting, right? We're waiting in anyway. The Orbitrap takes longer to operate at higher resolution. So if we're waiting anyway, we might as well run this at run the Orbitrap at higher resolution. And that gives us room to resolve multiple peptides in an MS2 spectrum. So this really just points to intentional precursor co-isolation and co-fragmentation, and then let the software tease out the different peptides. But we can do that because we're working in a high resolution mode with the MS2, and just in the nick of time, MSAID partnering with Thermo with their Proteome Discoverer 3.0 softwares developed and implemented this Chimera search engine that's really tailor-made to identify multiple precursors from a single MS2 spectrum. So you can sort of think of this as a hybrid of DDA and DIA, right? So DDA, we have these narrow isolation windows to try to fragment one precursor at a time. DIA, we have much wider isolation windows, and we're simply stepping through the MS1 range. And then wide window acquisition is basically data dependent acquisition, but we just broaden the windows. So we say, well, at least I'm going to get this one precursor that I centered my MS2 on, and I may get some additional peptides as well that are just hanging around there, but not directly targeted for fragmentation. So this is a project that T. Trong in the group is working on. And just to sort of step through what this looks like, this is just the number of MS2 spectra that we are acquiring as a function of the maximum injection time that we allow, and the corresponding resolution in the Orbitrap. So we can go longer injection times and higher resolution. So of course we're getting fewer MS2 spectra as we go to these longer injection times and higher resolution settings. So that's what we see as we step down here, and we're running the AGC target at 1,000%. So 10 times higher AGC target than what we're typically doing. And then we can look at the PSMs that we are acquiring. Of course, when we're doing standard data dependent acquisitions, not all MS2 spectra produce lead to a peptide identification. But now when we're doing this wide window acquisition with the Chimera software, this allows us to get multiple precursors per multiple PSMs per MS2 spectrum, up to 30,000 in some cases. And this is all with 0.2 nanogram HeLa, just replicate digest to develop the workflow. That seems like a good place to start. And so now when we look at peptides, remember we're at 30,000 PSMs, but a lot of those are redundant. So when we look at peptides, it's more like 10,000 peptides per 0.2 nanogram aliquot. This is still quite a bit better than about a 29% increase relative to our standard, or 28% increase relative to our standard data dependent acquisition. And it looks like the sweet spot is in this sort of 8 to 18 Thompson isolation window. across the board, regardless of resolution setting. And there are clear trade-offs, so it doesn't matter too much if we go faster but lower resolution, or a little slower and higher resolution. Seems like either of these areas work well. Now, when we include match between runs, the number, this optimization, we end up basically getting the same number of of overall peptide and protein identifications with these 0.2 nanogram aliquots, regardless of the specific MS acquisition settings. About 20,000 peptides, even with standard DDA, and about 3,500 proteins across the board. So this kind of begs the question, is it even worth it to run in this wide window mode? And really the high sensitivity is much more due to these crazy separations that we're doing than the isolation and data acquisition mode. But I think it is still worth it, basically doing the same sort of mixed species fact checking that Claudia talked about and that Ying talked about. We see about 4% of our peptides when we analyze these against a mix of yeast and human library. About 4% of our peptides are matching to incorrectly are matching to the yeast library, where these are FDR controlled at 1%. Our MS2 based identifications are FDR controlled at 1%. So I think increasing the ratio of MS2 versus match between runs identifications is beneficial, and that's what this wide window acquisition with the Chimeris software allows us to do. It also allows us to go faster. So we can go from 40-minute, and this is just 8 replicate analyses using a 12 Thompson isolation window and a 40-minute elution. You can see our MS2 identifications for this 0.2 nanogram aliquot, about 2,000 and about 3,600 with the match between runs. We can go to half the elution time and only lose 8 to 14% of the proteins that we're able to identify with this. So I think that speaks to a modest reduction in coverage for double the throughput is often going to be a worthwhile trade-off. We're also interested in comparing this to DIA. So this sort of summarizes just our general coverage with our 0.2 nanogram aliquots. So with 40-minute elution windows, with match between runs, like we said, it's not much different between standard data-dependent acquisition and the wide window acquisition. But again, our ratio of MS2 to match between runs identifications is really improved with the wide window acquisition. And DIA, this is library free and with a 10 nanogram spectral library here, the additional identifications that we see here. And then here we're going from 40 minutes to 20 minutes. And it looks like DIA especially really takes a hit in coverage when we go faster. Now, with this wide window acquisition, the replicate analyses, the principal component analysis between two different cell lines from K562 to HeLa is really outstanding. So you can see 62% in PC1, and this is 40-minute analysis of what is that? Or this is maybe 20-minute analysis of HeLa, 20-minute analysis of K562, and then 40-minute analysis of K562 and 40-minute HeLa. So really great separation in PC1, tight clustering, and then we get additional separation based on depth of coverage between PC in PC2 for the different gradient lengths. One interesting thing here, in parentheses, this is with library matching or match between runs, and without parentheses, this is with library free or data or no match between runs. And you can see we really do have better reproducibility in our assignments of protein abundance with the DIA than we have with wide window acquisition. I don't know if it's because this is more MS2 based and we're relying solely on MS1 for wide window and DDA, but that's just something interesting that we're seeing. And we have applied this to single cells. So these are HeLa and K562. You can see with the wide window acquisition, we consistently get a little bit better coverage with HeLa than we do with K562. And again, we're seeing a little bit poorer coverage for DIA. But on the other hand, we're seeing, again, a little bit improved CVs for DIA. Again, the PCA nicely separates. It's not so tight doing actual single cells, and that's a good thing because we want to look at and characterize this cell-to-cell heterogeneity. So if they all were exactly the same, there wouldn't be much point in doing single cell proteomics. So working with Sam Payne here at BYU, it was very convenient that two of us moved from PNNL to BYU the same year. This is looking at our old FAMES with 20 micron work. So basically to set this up, what we're doing is we're looking at the proteome coverage and the copy number as determined by Jesper Olsen's really deep dive into the HeLa proteome, so 12,200 proteins analyzed and quantified in a bulk scale study. And we're saying, well, what was the abundance of the proteins that we identified previously with our previous kind of best workflow from 2021, and now what is this wide window acquisition with the 20 micron ID separations enabling? And you can see that really we're pulling an additional order of magnitude in terms of the depth of the proteome that we can cover now. Still nowhere near what we can get with bulk, right? But an order of magnitude. There are entire classes of proteins that are not well expressed at these high intensity, high abundance values, and we can now see those by getting to over 3,000 protein groups per single cell with this, and then just doing a little bit of gene ontology. So this is looking at how many proteins per biological process or molecular functions we're now able to, what proportion of the Olsen group's bulk scale study could we see previously. You can see it was in the 10 to 20% for some of these processes. Now it's in the 30 to 40% of proteins are represented there. So I will go ahead and wrap up now. Just wanted to show that there have been still a lot of advances and a lot more to come in sample preparation, separations, and MS acquisition and data analysis. This is crucial for single cell and other low input samples. It's becoming increasingly robust, quantitative, and still not easy, but easier to perform. And now this wide window acquisition with 20 nanoliter per minute separations is allowing us to achieve a depth of 3,000 proteins per cell or aliquot in just 20 minutes. And of course, I'd love to thank a great research group that we have going here at BYU. Took a picture next to a dumpster. Sometimes we, say our group really closely resembles a dumpster fire, but still we're able to get some things done. Especially wanted to point out T here, who's done this wide window, ultra low flow stuff, and Xiaofeng is working on the multi-column LC system. I'd like to thank Thermo Fisher and some great collaborators there, Biogen, of course, Yingzhu was great to work with originally, and he's continued to be great to work with for the last 4 years since he became independent at PNNL and now Biogen, and our bioinformatics collaborators, and of course support from a variety of sources. Thank you for your attention. I'll be happy to answer any questions you may have.
Okay, thank you very much, Ryan. Really great talk, inspiring to everyone, showing that you could just go to the much shorter gradients and still reach the 3000 level of the proteome analysis, the single cell. I think that shows to the community that we go slowly but surely towards the RNA-seq numbers, but with much smaller CVs that RNA-seq people used to operate with. You do have a question from the audience. Let me read it for you. Did you use the same software, the data analysis of raw data obtained in the DDA, DIA, and then WWA mode?
Oh, that's a great question, and I didn't cover that. I should point out that this preprint is now online. And so we used Diane with MS Fragger for the DIA studies, and then we used Proteome Discoverer 3.0 for the data dependent and wide window acquisition. I'm not sure that there is one good software that can handle both DIA and these chimeric spectra. So we're pretty much locked in with the Chimeris algorithm when we want to look at these chimeric spectra with the wide window acquisition. But then Diane, and we haven't looked at Spectronaut, but we just chose Diane for the with and without spectral library for the comparison. And we're new to DIA, but we just knew that any reviewer would say, well, you know, okay, wide window is kind of in between these things. How did that look? So we wanted to start looking in that area.
Okay, thank you very much for your answer. If there is no any answers, questions, I do believe we come to the end of our today webinar. I would like to thank you very much for everyone who has participated today. First of all, for the panel of presenters, thank you very much for all your time. Being in very different, geographical scale and time, you've still been participated in this webinar. I think it would be very useful for everyone to see where the single cell proteomics is going to. All the participants, thank you very much for your participation too. I would like to hear your comments and maybe suggestions for the next type of webinar. The HUPO Single Cell Initiative plan to have another webinar like that in the springtime. That's why I would like everyone just to be preparing for the next round of it. And all the panelists, please suggest other people who you would like to see and hear on the next round of the webinar under the HUPO. And for those people who will actually be in the next HUPO conference, I would like to invite you on this Sunday. There will be a single cell discussion initiative that me and Claudia would actually be one of the people who will try to organize the questions that might come to the talks. So I please those who will be there enjoy it. Thank you very much for your participants, and hopefully see you later in the year.
Thank you. Bye-bye.
Thank you. Bye.
Bye-bye.